Good morning, everyone. Very soon we are going to commence the seminar.
Welcome to ARIBAS. Since the inception of ARIBAS in 2005, the institute has flourished remarkably, be it in academics or extracurriculum. My message for students is, try to be a man of success, but more crucial, try to be a man of values. ARIBAS believes in DNA, that is, do nothing average. So everyone out there, just stand by it.
Good morning, everyone. Very soon we are going to start inaugural session. We have with us our speakers, Dr. Hemant Soni sir and Professor Vandana Patrawale madam. We are waiting for our keynote speaker, Professor Pandyan sir. Sir will join very soon, within 5 minutes, and we will commence our virtual seminar.
Already we are late by 10 minutes. I think the invited speaker can join. We can start with prayer.
Yes, sir. Fine, sir.
Okay.
With due permission, we can start.
Okay, please start. Okay.
Nisha, as Udani sir has said, we can start with the inauguration function, and by that time the Pandian sir will also join.
Okay, madam. Fine, madam. Once again, good morning, one and all present here. Respected Engineer Shri Bhikubai Patel sir, President of today's event, Chairman Charutar Vidya Mandal and President CVM University, Dr. P.M. Udani sir, Chief Guest of the event and Director General Charutar Vidya Mandal University, Event Convener Dr. Bhakti Bajpai, In charge Head ARIBAS, Event Coordinator Dr. RItu Dixit, Head Integrated Biotechnology Undergraduate Department, Organizing Secretary Dr. Khushal Patel, Organizing Secretary Dr. Khushal Patel, today's distinguished speakers, Professor K. Pandian sir, Department of Inorganic Chemistry, University of Madras, Professor Vandana Patravale Madam, Department of Pharmaceutical Science, Institute of Chemical Technology, Mumbai, Dr. Hemant Soni sir, Associate Professor, Department of Chemistry, MS University, Baroda, all the judges of oral and poster session, faculty members, and dear participants. I, Dr. Nisha Dakshini, on behalf of ARIBAS, CBM University, would like to extend a very warm welcome to all of you to this one-day GUJCOST sponsored virtual seminar on nanotechnology, chemistry and biology at the interface of 21st century. ARIBAS and CVM University are highly grateful to GUJCOST for providing financial assistance to organize this national seminar. Charutar Vidya Mandal is the oldest and largest education trust of Gujarat, and CVM University is UGC approved well-known private university of the state. It offers undergraduate, postgraduate, and PhD courses in science, engineering, pharmacy, commerce, arts, and education. Aribas is constituent institute of CVM University, offering undergraduate, postgraduate, and PhD programs in pure and applied sciences like microbiology, biotechnology, genetics, botany, and zoology. Being an academic and research institute, Aribas always encourages students and faculty members to propose and participate in various research-oriented activities. The present virtual seminar on nanotechnology proposed with the objective of providing exposure in the field of nanoscale tools used to address unanswered questions in the interface between chemistry and biology. Before we formally begin the seminar, as per our Indian tradition, let's seek blessings from Almighty through a prayer.
हम 15-20 मिनट लागते हैं
What happened, Nisha?
समाहित करना प्रभाके टीकाति धन विभव सर्वस्व जनना नमूचु वन्दुचु विमल मुख स्वामी जगतना तुझी कायाथी घड़ित घड़ी वाणी थी उचरू इंद्रियोनी मुझ मन विषे भाव जस्वनु स्वभाव
Now I invite Dr. Bhakti Bajpai Madam, in charge head ARIBAS and convener of today's event for welcome address.
Good morning, everyone. On behalf of CVM University, I, Bhakti Bajpai, in charge head of ARIBAS, welcome each one of you in national seminar sponsored by GUJCOST on nanotechnology, chemistry and biology at the interface of 21st century. Most humbly, I welcome President of today's function, Engineer Bhakti Patel sir. His President, CVM University, though he could not join due to his prior engagement, but his blessings and best wishes are always with us. With due respect, I welcome Chief Guest of today's seminar function, inauguration function, Professor K. Modani sir. He is Director General, CVM University. He has always been a motivating and guiding light for us. Thank you, sir, for joining the inauguration session. My warm welcome, with utmost respect, I welcome Professor K. Pandian, sir. He will be joining very soon. He has been stuck in the traffic. So my most hearty welcome to the Professor K. Pandian, sir. He is today's first speaker of the seminar. He is from Department of Inorganic Chemistry, University of Madras, Chennai. I welcome other speakers of today's seminar. Second speaker of the today's seminar, Dr. Hemant Soni sir, Associate Professor, Department of Chemistry, MS University, Vadodara. Thank you, sir, for giving your consent and joining as resource person. Third speaker of today's seminar is Professor Vandana Patrawale, madam. Welcome, I welcome you, ma'am. She is from Department of Pharmaceutical Science, Institute of Chemical Technology, Mumbai. Thank you, madam, for joining, and I hope students will be benefited or enlightened after listening to your talk. I also extend my warm welcome to all the invited judges who will be joining these sessions later on, and they will be evaluating the oral and the poster sessions. So I thank all those judges for sparing their time in advance. My most humbly, I also welcome all the participants who have registered for this seminar. Around 180 participants have registered, and most of them, they are joining through YouTube link. And here I would like to specifically mention that we have seen very good response from the PG students. They will be presenting their dissertation work in the oral and the poster sessions. And being a PG institute, we always encourage and support such kind of enthusiasm from the students. So once again, I welcome you all in seminar, in the inauguration function, as well as the today's seminar, and hope it will be a good one. So have a nice day. Thank you.
Thank you, madam, for your inspiring words and welcome address. Our keynote speaker, Professor K. Pandian sir, have joined us. Sir has joined. And on behalf of ARIBAS, we welcome you, sir.
Yes, madam. Yes, madam. Thank you. Thank you. Good morning, madam. Good morning.
Good morning, sir.
Very good morning. Sorry for the delay.
It's okay, sir.
It's okay, sir. Sir, I welcome you in this inauguration function as well as in the seminar.
Yes, madam.
Thank you, sir, for joining.
Yes, madam. Yes, madam. Thank you, madam. Thank you.
Now I invite Dr. RItu Dixit, Head of Integrated Biotechnology Undergraduate Department and coordinator of today's event, to brief you all about the seminar.
Good morning to one and all present here. Honorable Engineer Shri Bhakti Patel sir, Chairman of Charutar Vidya Mandal and President of Charutar Vidya Mandal, University Vallabh Vidyanagar. Respected Shri Manish Patel sir, Vice President of Charutar Vidya Mandal. Respected Dr. P.A. Mudani sir, Director General, Charutar Vidya Mandal University. Dr. K. Pandian sir, Professor and Head Department of Inorganic Chemistry, University Madras. Principal Dr. Bhakti Bajpai Madam, our resource person, Professor Vandana Patrawale Ma'am and Dr. Hemant Soni sir, invited guests from GUJCOST Gandhinagar, faculty members, participants and dear student friends. On behalf of ARIBAS, I, RItu Dixit, welcome you all in one-day national virtual seminar on nanotechnology, chemistry and biology at the interface of 21st century, sponsored by Department of Science and Technology, GUJCOST Gandhinagar. Nanotechnology is an interdisciplinary subject, needs the knowledge of chemistry, physics, biology, and other sciences. Today's seminar on nanotechnology aims to orient indigenous research to a global trends to bring academicians, scientists, and pharma experts from all around the country together on a one platform to orient aspiring students of chemistry, biology, and other interdisciplinary subjects towards valuable research outcomes. In today's environment of discovery, there is no question that the researchers needs to be more versatile than ever before and be able to work across scientific boundaries. Hence, today's seminar will be an excellent platform to share and explore all the diverse and unique research findings. I am confident that from this seminar, our participants will be motivated towards interdisciplinary research. I am happy to share that out of 200 participants, more than 116 researchers took part in oral and poster presentation. I am sure to the participants that they will enjoy all the sessions. I would like to thank GUJCOST Gandhinagar for entrusting our institute with financial assistance to conduct this seminar. Lastly, I would like to thank Dr. K. Pandian sir, Professor Vandana madam, and Dr. Hemant Soni sir for accepting our invitation in spite of their busy schedule and grace the event. We are highly obliged, sir and madam. Lastly, once again, I would like to welcome you all for the seminar. Thank you. Thank you very much.
Thank you, madam, for this detailed information. The president of today's event is Engineer Shri Bhakti Patel sir, Chairman Charutar Vidya Mandal and President CVM University. Sir has always extended his kind cooperation and support for organizing such a technical events. We are highly thankful to him for the same. Due to unavoidable circumstances, sir is not able to join us. I'm going to read the message sir is conveyed. It gives me immense pleasure in knowing that Ashok and Rita Patel Institute of Integrated Studies and Research in Biotechnology and Allied Sciences, ARIBAS, has organized GUJCOST-sponsored one-day national virtual seminar on nanotechnology, chemistry and biology at the interface of 21st century. In today's world, there is a constant need to keep one updated, and this is possible only through innovation and research rather than chronicled knowledge. Such seminars and conferences are excellent ways of exposing the students to better and newer ideas. It also acts as a catalyst for the research students as they can put forward their research work and ideas before a wider audience. I'm sure that this virtual seminar will benefit all the participants. I congratulate all the persons involved in the seminar and I wish it all the success. Thank you, sir, for this inspiring words. The chief guest of today's program is Dr. P.M. Mudani, sir, Director General, CBM University. I would like to request Dr. P.M. Mudani, sir, to address the audience. Sir, please.
Good morning, all.
Good morning.
On behalf of CBM University, I once again welcome you all, my faculty team, then invited speakers. So everything cannot be learned in a classroom. That is very clear. So classroom teaching can only serve 50%. Rest of the things student has to learn through this kind of seminars, field trips, industrial visits, and association with so many institutions and industries. Now coming to the topic of the today, so India had done wonderfully well in a space technology or nuclear technology. The reason is the space technology was started by Vikram Sarabhai some 60-70 years back. Same thing was Rajaramana started a nuclear technology and then also as a legacy of 70 years. But nanoscience was started in India very late. So similar kind of achievements are not there as of today. If I remember exactly, nanoscience mission was started in India in 2007. Budget allocation in 12th plan was also very good, but somehow we do not have enough number of scientists who can carry out research in nanoscience. So that is a lacuna, and this kind of a seminar and workshop will definitely motivate the young minds to contribute in a nanoscience technology. Let me give a story of a few countries who have done wonderfully well. China, Taiwan, South Korea, they could understand the importance of nanotechnology and they specialize in a memory devices, display devices, computer technology. And today they are leaders in entire world. Even US cannot touch them, Taiwan or Singapore or Japan. They concentrated on memory technology, concentrated on processing power, concentrated on display technology, and they have done wonderfully well. China did other way around. They could see the business opportunity. Once this nanotechnology will pick up, people have to do research at molecule level. So where are the instrumentations available? So China focused on instrumentation technology. So today, across the world, maximum research instrumentation is provided from China. So we have done our job. DST is pushing very hard from central government. State government, science and technology department are also doing wonderfully well. But still we have a miles and miles to go ahead. Our journey just started 10-15 years back and we have not achieved the milestone which can be recognized across the world. The reason may be we had a limited budget and our focus was too much diverse. We wanted to do in agriculture, we wanted to do in a textile, we wanted to do in many areas. So as far as ARIBAS is concerned, we have three focus areas: industrial biotechnology, medical biotechnology, environmental biotechnology. So for these three areas, if you are able to ignite young minds, definitely this kind of workshop will ask them to do some sort of research. Fund is available. From state government, DST department fund is available. Central government, DST department fund is available. There is a special mission also on nanotechnology, nanoscience. And you can refer to the institute which is there in Mohali, Chandigarh, in Nanoscience and Technology Institute, Mohali, functioning very well. So my message to the young students, or most of the Bhakti said that most of the students are from PG department. So definitely this kind of seeds must be planted and they must be oriented for the research work. And now we have started a PhD program at our own institute, and a few bright minds are also invited to join whenever opportunities are there for our PhD program. So I wish great success and all invited speakers having experience from Madras University, MS University, Institute of Chemical Technology. These are all renowned institutions, and whatever experience they have, definitely they will be able to share within limited time, whatever possible, and should be igniting the mind so that they can do the research work. So once again, I congratulate Bhakti and all ARIBAS team and wish a great success for this today's online seminar. Thank you.
Thank you, sir.
Thank you very much, sir, for your encouraging words. Before we begin today's technical sessions, there are some instructions. Each expert session is of 1 hour, followed by an interactive session. If you have any question during the presentation, please type them into the YouTube chat box. We'll bring them up during the seminar, and we'll also have time for questions at the end. Now, I would like to invite Dr. Digvijay Rana to introduce our keynote speaker of today's seminar, Professor K. Pandyan sir, Department of Inorganic Chemistry, University of Madras, Chennai, Tamil Nadu. Rana sir, please.
Good morning to all. It is my great honor and pleasure to introduce Dr. K. Pandyan sir. Sir is Professor and Head of Department of Inorganic Chemistry, University of Madras. Sir obtained his PhD from University of Madras in 1995. Then he joined National Geophysical Research Institute, NGRI, of CSIR lab at Hyderabad as a scientist B. Then he went for postdoctorate at Taiwan, during 1997 to 2000, and since 2000, he is working as a lecturer in the Department of Chemistry, University of North East. And currently he is working on nanotechnology, fuel cell, sensor and imaging. And his achievements have been recognized by many different awards. He is recipient of 10 Best Paper Awards in national level conference. Sir received Best Research Award in 2017 as well as 2018. Sir received Best Scientist Award of the year 2018 from NASA Foundation, New Delhi. Sir is member of several national and international societies. Sir completed more than 17 projects and published more than 180 research articles. So it is such an honor for us to have Dr. K. Pandian sir with us. I welcome sir to enlighten us with his keynote address on the topic of rapid detection of bacterial and viral infection. Welcome, sir.
Thank you, sir. Thank you very much for all the participants and also the organizer. So really apology for this inconvenience and also delay. So I thank this organizer, Professor RItu Madam, and also Professor Udani, and also Professor Rana, and other faculty members for this nice event and also wonderful arrangement. Slide visible, madam?
Yes, sir. It is visible, but we can still full screen it.
But I don't know the options because
Now, sir, screen is also not visible.
oh, sorry, madam. Okay. Just a minute, madam. Just
Yes, sir, it is visible now. Presentation is visible.
Okay, okay. Thank you, madam. Thank you. It's okay now?
Yes, sir, fine, sir.
Okay, madam. So good morning, all of you. So I'm going to maybe talk about some of the aspects of nanotechnology for sensing and also screening of some of the bacterial and viral kind of diseases. So as Udani sir mentioned that India started very late, maybe 2007. But in my case, I started my career in 1997 in Taiwan. So there I learned all the nanotechnological issues from basic to from the applications. I'm going to highlight some of the important findings and also our contribution to the society. Many students maybe the more involved for this kind of research for application point of view, especially for recent days that the viral infections, one of the dangerous issue for human being. So this is some of the highlights of my talk because colloidal gold particles are brilliant color, so that is easy to functionalize with the various receptor molecules like mannose and other some of the drug molecule over the colloidal particles. Colloidal particles are brilliant color. So because of the aggregation with the bacterial organism, there is a color change from the pink color to blue coloration or colorless indicate the formation of bonding with some of the microorganism. And also we can use some of the magnetic nanoparticle with the Khushal kind of functionalization with some of that receptor molecules. So we can see this kind of this is easy method to preconcentrate some of the bacteria available in the water sources. We can increase the concentration of bacterial level and also easy to eradicate. And also this method we are using for magnetic nanoparticle for the removal of some of the biofilm, some of the bacterial infections. And also we are also working on small peptide molecule nowadays. People are using as an antibacterial agent. So we can use it to functionalize with the thiols or other functional group. So conjugate with the metal nanoparticles we can use for antibacterial applications. And other topic is we are going to discuss about some of the photothermal studies. I'm going to start this work now. One student is joining the group. She is going to concentrate on photothermal effect to eradicate some of the bacterial infections. And also we can use some of the conjugated drug molecule with the gold or silver nanoparticles. So this will help us to eradicate some of the maybe multi-drug resistance microorganism, especially for that kind of combinations so highly working. So we are concentrating this kind of drug conjugated gold and silver nanoparticle for multi-drug resistance organism. And also we can use some of the immunoassay method to conjugate with some of metal nanoparticle for antigen antibody binding mechanism. We can quantify or maybe screening of some of the microorganism. So because chemistry background, we can do a lot of wonders, and also we can do a lot of studies with the help of chemical functionalization, some of the drug molecule or polymer. So we have enough experience in handling of metal nanoparticles, easy to prepare and also easy to functionalize, and then we can apply for some of the applications. So this some of the causes of microorganism. So infected diseases like a major contribution for the human mortality and also pose significant global impact to the mankind. So bacteria leads to that fatal infections and diseases caused by the contamination by the food and also water sources. And this is an alarming issue for human being. And also the earlier maybe detection or maybe diagnosis of these diseases, one of the important issue. And also conventional method, people are concentrating on detection of colony specimen, including maybe it takes some time and also maybe a time consuming process. So people are concentrating on other technique like PCR and also immunoassay method like ELISA method. People are concentrating to quantify fast detection of this bacterial infections. So this another issues one of the people are come across nowadays caused by the continuous intake of antibiotics for very diseases causes a multi-resistance organism. This is called a mechanism of antimicrobial resistance. So because this not easy to eradicate this bacterial kind of infections. So we need some kind of alternate method to kill this microorganism. So we are concentrating on many aspects. Especially I am going to discuss some of the important issues. So this, as you know, this is a list of microbes caused by various diseases, especially for bacteria, fungus, virus, and also protozoa. These are the main causes for various infections and also human threat. So this, especially for the bacteria, we create some of the ulcer, the thyroid fever, and also some kind of cholera, and also the other diseases, bacterial diseases, and also the fungus. One of the culprits for Aspergillus candida alfonso, that create some kind of fungal diseases. Apart from this one, we have the viral infections. Recently, we have come across COVID and other diseases, and also this other dengue virus. These are the one of the major issue for human being. And also some of the protozoa. This is living in the stomach and also the contaminated with the water sources. This creates some kind of fever and also some of the infections. So these are the major diseases we come across in the day-to-day life. So how to quantify and also how to find out this kind of pathogenic sensing. So we have various methods, especially for we use culture method to find out this physical appearance as well as chemical assay to find out what kind of microorganism present in the water sample or maybe food sample. And also we can do some kind of molecular methods, for especially for we use polymerase method, PCR, and also DNA micro approach, and also fluorescence sensing, and also some kind of peptide sequence analysis. Also, people are concentrating to quantify this detection of microorganism. And apart from this one, we are concentrating also the immunological method, especially for like antigen antibody binding method. People are concentrating on easy method to quantify very low concentration also micropresent in the water sample, other body fluid. So these are the method mostly we use for sensing of some of the pathogenic organism. So this is coming to this slide. You know, this slide is gold colloidal particles are billion color. This color is some of the naked eye can see the color changes because of this brilliant coloring nature. So this gold nanoparticle can be easily functional with the thiol molecules with the various function group. So here I shown some of the this functionalized with the gold nanoparticles. So once it is react with some of the microorganism or maybe viral organism, so this is forming some kind of interactions because of aggregations or maybe chemical bonding, or maybe it is releasing from this the receptor molecules getting aggregated, or maybe there's action for etching. So this kind of sensing, this kind of coloring nature can be visualized with the colorimetric method. Also, we can use for fluorescence method, light scattering method. Also, we can use, and finally, we can use for SERS method as a surface enhanced. Maybe Raman technique is one of the simplest technique to quantify some of the microorganism. And this is a concept we use. People are using. So gold nanoparticle is used to functionalize with the various molecule, especially for peptides, and also the antibody, and also aptamer. Aptamers may be nothing but oligomeric, some of the DNA molecules. We have to selectively prepare that some kind of binding nature. We selectively bind with a particular organism or something else. So that method is one of the interesting method. Recent days, many people are concentrating on aptamer based research. So this advantage of this aptamer can be recycled and also regenerated from the once application of any sensing method. We can regenerate that aptamer. So though it is expensive, but this repeatedly we can use this kind of aptamer technique to find out this various kind of infections. This once it is forming react with this bacteria, this can be quantified with the search already told you, and also some of the fluorescence enhancement, also some of the charge transfer method, and also we can use for colorimetric method. This is a color change from pink color to blue color. This is easy to find out by naked eye itself. That is the advantage of this method. And this is coming to this slide. This is an interesting slide. The microorganism containing this cell wall, where the amino acids like some of the dipeptide, alanine, alanine. So this is the cell wall. The amino acid is to react with some of the vancomycin kind of antibiotic. So vancomycin, one of the antibiotic people are using for some of the urinary infections, and also very expensive. So to avoid the cost effective, we are doing some kind of methods to quantify this kind of the bacterial infections with the help of some of the nanotechnology approach. So this is a slide. The first one appeared in the literature very long back. So this is the one that people are using. Vancomycin conjugated some of the mercaptan thiol molecules. This with the modified with some of the gold particles. So this will attack into the microorganism, bacteria, forming some kind of interactions, then the kill that kind of bacterial infection with the help of this vancomycin kind of drug molecules. So like that, people are concentrating on the conjugation with some of the drug molecule. It is easy for you to identify as well as some of the to kill this microorganism with the help of compact effect. So like that, people are concentrating on some of the drug molecule conjugated with the gold particle through covalent kind of interactions, very strong bonding. So this is a way we have also developed some of the method for functionalized gold particle using amino glucoside kind of antibiotic like gentamicin and also neomycin. We have developed a method, and also we have a couple of articles published in recent journals. And the other method came recently in this literature. If you look at the literature, there is another article recently came. Here they use without any functionation of this vancomycin. So because the vancomycin contains a lot of function group, we have this carboxyl group and also the acid group. Easy to form a bonding with the colloidal gold particles. These people are concentrating on institutive method of synthesis of auriculoid, disposing auriculoid, then adding this vancomycin molecule to this, maybe the a single compartment. After addition of this reducing agent, it is forming the colloidal gold particles. This vancomycin act as a capping agent to stabilize the colloidal particles. So by this way, we can prepare some of the drug molecule modified colloidal gold particles. So they utilize this kind of nanocomposite for the killing of multi-drug resistance microorganism, especially for Pseudomonas aureus. They use this kind of composite material for killing of microbes. So they got very good result, and also you can see this colloidal particles absorb around 5 to nanometers. This is a plasma band vibration. So once it is forming aggregate, so this is a mechanism how it is the react with the auriculoids, and also how it is forming complex. Then the alkali medium it is forming the colloidal gold particle. The particles here this vancomycin act as a capping agent to avoid the aggregation of the colloidal particles. So like that, the people are prepared and also they exposed into various microorganisms. The vancomycin modified colloidal gold particle into various microorganisms like Pseudomonas aureus and some of the E. coli and other microorganisms. They can see this kind of control that controlling of infection level and also the curing of microbial infections. And they confirm with some of the electron microscope observations. Once it is how this microbes getting infected with this kind of nanocomposite, how we can eradicate this thing explained in the slides. And also this when they find out this, they compare this bare vancomycin along with this the gold nanoparticle modified vancomycin. They found out this OD values, optical absorption values, by this kind of a composite, and they reported that the composite containing the very low value can kill this microorganism, and also effectively can be used for instead of using the vancomycin. Thereby, can reduce the cost effect of this vancomycin kind of drug, and also this completely remove eradicate the infection of this kind of microbes. And this is values are given with the different kind of microorganism like Pseudomonas aureus and also E. coli derivatives and some of the E. coli strains. And they reported the results the compare with the bare vancomycin as well as the composite kind of vancomycin capped gold nanoparticle. It gives a very low concentrations enough for killing of this microorganism. So this you can see the concentration level of micro the drug molecule used for this eradication of drug molecules. And once it is functionalized with the vancomycin, we are getting the very low concentration. Also, we can kill this microbes. That is the advantage of this method. And also, we can avoid this kind of overdosing of drug molecule for killing of some of the urinary infections. And this is other case. The people are concentrating on colloidal gold particles, colloidal gold particle. People are concentrating to use colloidal gold nanorods. So here also you can use to kill this the E. coli kind of microorganism. The people are using this gold nanorod. You can see this color changes from the billion color to blue coloration indicates the reaction with the microbes. So similarly, we can do a lot of studies with some of the thiol molecule functionalized colloidal gold particles react with some of the microorganisms leads to the aggregations. From this, we can quantify the level of infection and also the concentration level of the microbes present in the water sample or maybe in a serum samples. So like that, people are other studies like the salmon kind of microbes. You can use this maybe enhanced Raman spectrum to quantify the amount of microbes present in the some of the food sample as well as that serum samples. So like that, we can also use some of the like in the other studies. The people are they use various kind of conjugated system for the killing of microbes and also pre-concentrate the microbes. The kind of studies. So this is one of the recent studies. People are concentrating on photothermal approach to remove some of the microbes using some of the photosensitive color dyes like rose Bengal and yosin dyes can be used. So this is the dyes we can easy to absorb light energy, laser light or maybe as a LED light. Then it is react with this microbes and also can kill this microbes with the help of photothermal kind of slices. So like that, you can do a lot of studies with the help of a simple technique can be used for so killing of microbes. And this technique extended to further, we can use for high throughput screening of number of samples put it together, and we can do fast screening of this infection and also killing of microbes with the help of high throughput screening, and also they can use for microarray plate reader for counting of the bacterial infections. So like that, people are concentrating an advanced method and also very easy method to find out. So the infection level also you can see this extent of infection can be quantified with the help of this kind of studies. And this is recent studies. People are concentrating on some of the nitric oxide kill that microorganisms like Pseudomonas aureus. One of the nitric acid in one of the dangerous molecules that disrupt cell walls of the Pseudomonas aureus kind of microbes. We can see this kind of the fluorophore conjugated with this kind of and the kind of molecules that are easy to functionalize with the kind of simple molecular system. So once it is maybe removed by radiation with the light, there's once the maybe detached in this molecule, we can see the color changes and also fluorescent color. We can monitor the some UV light radiation. We can see the color changes like that. We can quantify so the level of infection as well as simple technique to find out this Pseudomonas infections. Another technique, people are concentrating some of the layers such as double hydroxide, like lithium aluminum hydroxide kind of molecules layers such as can be used for killing of microorganism. So in this technique, people are concentrating on layers such as LDH kind of layers such as nanostructure materials. So once they use exposed with the silver nitrate, and then the glucose is here they use as a reducing agent. So this is some of the layers such as molecules that the nanostructure can be modified with some of the slow nanoparticle. Then it is functionalized with some of the Van kind of drug molecules to make a conjugate. The conjugate can be used for killing of some of the microorganism and also to find out this extent of infection as well as so the incubation time also you can find out from this kind of methods. So like that, people are concentrating various methods to find out this kind of eradication of some of the so this microbial infection as well as dangerous biofilm and other kind of residual kind of microbes. So this can be visualized with the help of optical microscope to find out this kind of whether this Pseudomonas aureus or E. coli, whether it is alive or dead with the help of some of the dye molecules, staining agent. Like that, people are concentrating on more on this kind of nanostructure materials, functionalizing some of the vancomycin and silver composite can be used for screening as well as killing of microbes. And this other method, like they use monos selectively bind with the bacterial cell wall. So people are functionalizing this maybe some of the thiol molecules or some of the amine molecule functionalizing some of the manos. So this is attached with some of the magnetic nanoparticle like the core shell kind of configurations. So this also can use for to pre-concentrate some of the microbes and also can quantify the infection level of microbes. So and this is method, the similar method they use for bacterial screening. So they use some of the magnetic nanoparticle functionalize some of the antibody antigen. So from this we can quantify very trace amount of microbes present in the water sample or maybe other maybe the food contaminated samples we can quantify with the help of immunological approach. So they emitting some light. You can see this fluorescent immunoassay method is helpful for finding the lower concentration of the microbes. And also the people are concentrating on the light control method for effective killing of potassium approach. They use polyaniline. So this is one of the important polymer, very less price, is available everywhere. You can use this kind of polymer modified substrate to quantify this bacterial level under the photothermal approach. So then also they do a lot of studies to confirm this infection level, and also so this killing of microbes with the various kind of optical and microscope studies, and this they use AFM and other technique to quantify the level of infections. And they measure the colony level with the help of maybe UV visual studies. They quantify the infection level, and also the concentration level also optimized with the help of OD studies. So like that, the people are concentrating on polymer kind of a coarse kind of nanoparticle for sensing and imaging of bacterial infection with the help of different microorganisms, and also screening with the help of some of the fluorescent light sources can be used. So and other things, other method like some of the peptides, already told you, the peptides are showing some kind of antibacterial effect. So it is easy to conjugate with the EDC method. We can functionalize with the thiols or maybe a phosphine group. That can be modified the surface of the colloidal gold particles. So this forming covalent bonding over this colloidal particles. So it is easy to maybe this immobilize the peptide molecule over the colloidal particles through the covalent bonding. So that can be exposed into some of the microorganisms, then can be quantified the sub-microgram concentration level of bacterial infection. And this is a mechanism that also they find out this type of conjugations before and after conjugated with some of the peptides. And also this microscope method we can use for maybe electron microscope can be used to quantify this colloidal particle, as well as making this kind of peptide modified molecules can be quantified with the help of electron microscope. And because the peptides are optically active, we can confirm this nature of binding with the nanoparticle can be visualized with the kind of cyclone diagram kind of maybe some of the uh, spectroscopic analysis, and also we can use the fluorescence microscope to visualize the nature of bonding over the colloidal gold particles. So and also we got to optimize the pH and the other parameters to so enhance this kind of fluorescence emission from the peptide modified colloidal particles. But these are the various studies to measure this extent of infection, and also so quantitative analysis people are concentrating on the fluorescent intensity variations. They read out by the fluorescent microscope and also confocal microscope. We can use the normal SEM as well as electron microscope to find out so the killing of microbes, and also the live cell with the help of some microscopic analysis. And this some of the studies are recently reported in the literature. We can use people also concentrate some of the natural clay. This is a halloysite. Halloysite nothing but a natural clay. So it's available in the market. So this is like some of the nanosachet, some of the nanotube kind of organization. So we can see this kind of outer surface can be modified with this kind of some of the antibodies over the surface. Then it is reacted with some of the microbes, and they're forming some kind of interactions that can be quantified with the help of the antibody antigen binding kind of interactions. So this is methodology given here how to functionalize this antibody over this halloysite surface. Halloysite nothing but a natural clay. The clay can be functionalized with some of the amine molecules, then followed by the EDC coupling method. We can attach this kind of antigen antibody binding studies. We can visualize this functionation process with the maybe FTR and other technique can be used, and then we can expose into some of the bacterial organism. We can see the kind of infection, and also what type of microbes also can be visualized with the help of these studies. So like that, people are concentrating on various nanostructure materials, gold nanoparticle, quantum dots, and also fluorescent dye modified magnet nanoparticles, and also some of the dye modified silica can be used for visualization of some of the E. coli, and also salmonella, and also pseudomonas aureus, and other maybe tuberculosis kind of diseases. So many people are concentrating on this kind of simple technique can be used for visualization of some of the microbes, as well as can be screened with the cells and also fluorescence, and also sometimes we can use for FTR and MALDI method can be used for quantification of this other infections. And coming to this fluorescent kind of quantum dots, we are also concentrating. We got some project from the DPT for imaging of cancer cells. And this is a concept we use in the fluorescent dyes are very stable coloring agent compared to color dyes. Color dyes is nature of colors once exposed to light, the color fading quickly. To avoid this kind of degradation of dyes, people are concentrating on to use quantum dot. Quantum dot is a brilliant color and also a very sharp intensity. So this highly stable emission getting from some of the quantum dots like cadmium telluride and also cadmium selenide over coated with the cadmium sulfide kind of Khushal kind of quantum dots. So this is a concept that people are concentrating on this conjugation of the quantum dot. So this can be this is this can be synthesized in the chemical laboratory. Carbonic acid functionalized the quantum dots. So we can use some kind of the bonding with the EDC coupling. We can couple with some of the biomolecules or receptor molecule over the surface. So whether we can do the covalent bonding or maybe replacement reactions, and also electrostatic interaction, we can bind with some of the fluorophore, some of the receptor molecules, and also we can do with the antibody antigen. Also we can do with the EDC coupling method. This kind of receptor molecules is easy to find with the microbial and as well as viral components. You can use it to screen this kind of method with the various kind of screening technique. So this is a concept that people are concentrating on this coastal nanoparticle. So this is the core and also outer shell. So this is inorganic shell and also the organic sheet outer capping agent. So here they attaching some of the receptor molecule. For cancer studies, people are using maybe transparent and also this folic acid as a receptor molecule. The idea of using this receptor molecule, this attack only the cancer cells, not attacking the normal cells. So that way we can easy to visualize the cancer cells with the help of this method and this kind of mechanism. And this is one people are concentrating on how to use this kind of lipoic acid, one of the organic small molecular molecules. It is easy to functionize with the various kind of polymer as well as small molecules. So here they use the chlorinic acid functionalized quantum dots with some of the thiol exchange reactions followed by EDC coupling method. So and this conjugate of this polymer can be used for to find out this microbial infection as well as viral infections. Other people are concentrating on some of the maybe lactose and also mannose functionalized the chitosan. So chitosan also one of the biopolymer is easily available. So we can do some chemical synthesis method. A simple method we can do with a synthetic approach, a simple 1 or 2 steps. We can use it to functionalize some of the receptor molecule over the polymer surface. This polymer acts as a capping agent. We can use it to functionalize this quantum dots, Khushal kind of quantum dots, so functionalize some of this chitosan based polymer. So it can be used for visualization of some of the yeast cell and also microbes present in the water sample and other samples, food sample for screening of the infections. And this is some of the contribution from our laboratory we are concentrating on. So some of the biofilm as well as we have done some studies for silver highlights for the extracted from the plant origin. And also we have developed some method for antibacterial agent of aminoglucoside conjugated nanoparticle for killing of microbes. And also we developed some of the quinoline kind of antibiotic. This containing NH group. NH group is to functionize the dithiocarbamate or maybe SKG group. So this paper also submitted, we published long back. Recently we got one paper from the Electrochemical Society. They use in that paper we use one of the ATP, one of the biomarker for bacterial infection. So we can quantify the ATP level in the water sample or maybe food sample. That is an indirect way to find out this kind of microbial infections. And similarly, we can use recently we are also concentrating on screening of some of the viral infections. Like already is available, the market is available. So we follow this method for screening of some of the blood sample with the antigen antibody mechanism to find out this kind of what kind of viral infections. So here the people are using some of the nitrocellulose paper as one of the substrate to immobilize some of the antibody and antigen. So from this kind of once exposed with some of the serum samples and forming some of the color changes. From the color changes, we can selectively binding the molecule interact with the selective binding of antigen. We can screening particular viral infection. Especially we are concentrating on dengue and other diseases. Now we extend this research into COVID also we are testing now. So COVID test, they very well. This is some of the method people are follow. So this is we regularly use this method for a collection of this sample from this from the nose and also then maybe lies with some of the buffer. Once getting this kind of some of the maybe the RNA molecules, RNA can be visualized with the help of colloidal gold particle, functionally some of the receptor molecules. So the bottom one they explained how this the viral particles react with the colloidal particles. There's a color into blue color. So this indicate the formation of some kind of interaction with the some of the viral infections through the viral RNA molecule easy to functionalize with the colloidal particles. So like that we can easy to functionalize and also visualize the colloidal particles with the help of simple technique. So this also we extended for naked sensing we are using some of the nitrocellulose kind of paper to anchor this kind of colloidal particles. Then we can expose into some of the RNA molecule collected from COVID cases. We can easily screen by naked sensing itself. So this is the concept how this the viral RNA is collected and also then how it is reactive with the colloidal particle receptor molecules and how it is forming aggregate. Then how we are quantifying this kind of color changes. Another technique people are concentrating on this viral colloidal particles bind with the antigen. So once this exposed into some of the viral particles, virus, it is forming some kind of aggregate. So this aggregation can be quantified with the help of some of the light scattering method, the simple method to find out the extent of infection level of COVID. So this is a simple small molecule react with this colloidal particle. We are not getting any aggregate. So once in this attachment of this some of the target molecules is to form aggregate. So higher concentration of some of the target molecules, we are getting this bigger size particles that can be visualized with the help of some of the light scattering approach to quantify the extent and the concentration level of viral infections. So and also we can use a different kind of particle, gold particles, maybe quantum dots conjugated with the antibody. So we can visualize with the color changes by naked eye sensing approach. Can quantify this kind of methods we can call this the rapid test method. So we are concentrating on this kind of research. We are expecting some kind of results. Maybe we are succeed this kind of ideas. And similarly, we can use a different kind of infections in the same slide. Can use a different viral testing kits. Also, we are concentrating. So because this is one of the emerging area research, and also we have trained nearly we are working 2 decades. We are concentrating this kind of handling of nanoparticle for various kind of applications. This is our collaborators. Professor Utaita lab only trained this kind of nanotechnology in 1987 to 2000. So there I got the opportunity to work with some of the nanoparticles, and also we got many applications from his lab. And also other people also now we have collaboration with the Taiwan, Malaysia, and also Iswaramurthy, one of my friend is working in here, by Bangalore JNC sir. He is supporting us to help for some studies, and also we are concentrating on agriculture research and also the veterinary college also we are doing research. So like that, these are many outcome from coming from our research scholars, JRF and also MTech students, MS students. So I thank all of them for this their valuable contribution. And some of the fundings we are getting from UGC 3 times we got this project funding UGC for sensor and also liquid studies. So I got Young Scientist Award from the DST, and also we got the Nano Mission some of the controlling of some of the viral infections and also nanomedicine. We got funding from DPT for quantum dots conjugated system for imaging of cancer cells. So and also we got many grants from this ICMR and also this SERB kind of funding agency. So these are the funding agency we received for various kind of applications. So he is my professor boss. He is from Taiwan, Taipei. He is the middle one from IIT Madras. Dr. Pradeep came to our lab. We had developed the system the initial period in maybe 1988. We have established this kind of method for the handling of nanomaterial for various applications. So these are some of my research scholars passed out from my lab. The first one is called Nirmala Grace. She is now working in the VIT as one of the head for this division, nanotechnology division, and head in charge. And also some of my students are working in a college and also other places, universities. One fellow in a postdoc is doing a South Africa is working for his postdoc. He is the one that developed the quantum dots for sensing and screening of various cancer cells. Then the first one established is this antibacterial activity of aminoglucoside kind of antibiotic for screening of bacteria and viral infections. So these are my currently we have many students in my lab. They are also concentrating on various research work, especially for we are concentrating energy and environment as well as biological applications. So maybe this last one is working for polymer conjugate some of the antibiotic for screening of drug molecules is working now. So I thank all of you for this active participation. Sorry for the delay, and also this we have other commitment because of that one we unable to produce more number of slides and also some of our article also I wanted to show you in front of you. So suppose any students interested can come to our lab and also can contact me through email. I maybe try to help you and in all aspects. So this is a simple method and also so more advantage for each one. Hello.
Yeah, thank you very much, sir, for such a nice session.
Yes,
sir.
madam. Yes, madam. Sorry, madam. Sorry for delay.
Oh, sir, it's okay, sir. So,
Any
thank you
questions
very
from audience, madam?
Yes,
Please.
The session was actually interesting and informative. I'm very sure participants get benefited by this
Yes, madam.
informative
Yes, madam.
session.
Yes, madam.
Now session is open for discussion. We have taken some raised questions.
Yes, madam. Can we read out and answer, madam? No problem.
Okay, fine, sir. Sir, there is one question.
Yes, madam.
Which method is more reliable for the conformation of nanoparticles synthesized?
Chemical method is easy method. You can use nowadays glucose can be used as a reducing to synthesis of gold particle. Even we can use ascorbic acid. Simple technique we can people are following. I can send some paper also to madams. Again, people can work in that kind of aspects.
Basic methods, yes, sir. Fine, thank you, sir. Next question is, which other metals can be substitute for gold?
We can use
metal.
That advantage of gold particles are stable one. That is why people are concentrating gold. The other nanoparticle, maybe silver, we can use. Silver itself it shows some antibacterial activity. Okay. And the copper also can be used. Both the maybe silver and the coppers are mostly unstable. Stability is very less. Yes. And also synthesis also not easy to synthesis. People are reporting, but getting uniform size is difficult to uniformity. That is a drawback. So my advice is people can use gold nanoparticle because other nanoparticle like palladium and all colorless. Platinum also some of the black color, especially color black in color. Okay. Not showing any plasma response.
Correct. Fine. So it is more stable and we can also
yes. It easily. And easy to functionalize also. Functionalization is also easy, madam.
Yes, sir. Yes, sir. Yes, sir. Fine. One more question. Can these gold particles used to detect fungal infections?
Yes, madam. Yes, madam. I can use because ketoconazole one of the antifungal agent. Okay. So we can make some kind of ketoconazole. We can use some of the polymer capsule. So I have a lot of studies. I can show you some slide because of the time delay. So I can show you that slides because we can use polymer conjugated the ketoconazole can be used for treating of some of the fungal infections.
Fungal infection. Yes, fine. Is there any other question from audience? Raj Naik wants to ask one question.
Yes, madam.
Just a minute. We have prepared curcumin-loaded carbon dots using hydrothermal methods
Yes.
for antimicrobial applications.
Yes.
However, we do not get uniform-sized carbon dots, and even accumulation of carbon dots is a problem. So, what can be done to avoid such accumulation?
So nowadays people are concentrating carbon dot. Carbon dot because easy to prepare and also and thing is you got to handling is very important. Handling how to purify the carbon dot. We have to use some of the dialysis method to purify carbon dioxide with get a uniform size. So that way we can control that the emission also can be tuned by dialysis method, and also easy to functionalize with some of the carbon group, and also curcumin is a well-known method. I can send you some paper also if they interested. They can ask me through mail. I send some papers also.
Yes, sir. Yes, sir. So I hope Raj, your doubt is solved, and even we can contact sir through mail if we will have any
Yes,
doubt.
madam. Yes, madam. Always welcome because we are student friendly. We are working for the students. Without students, we can run the show.
Thank you very much,
That is
sir.
my
Thank
aim.
you.
Okay, madam. Thank you, madam.
Is there any other question?
Any?
Yes, Hemant Soni sir has raised
Yes.
hand.
Yes,
Yeah.
yes, sir.
Good morning, sir.
Yes, sir. Good morning,
Good morning,
sir. Good morning, sir. Good morning.
Yes, your work is very interesting.
Yes, sir.
Particularly, you are anchoring the
Yes, sir.
vancomycin.
Yes,
You are
sir.
anchoring vancomycin on the surface of these gold nanoparticles.
Yes, sir. Yes, sir.
And then this gold nanoparticle transpect a bacterial cell.
Yes, sir. Yes, sir.
Okay, and then the cargo or drug is released inside
Yes,
the
sir.
bacterial cell.
Yes, sir.
And then after all, the question is that what is the fate of this gold nanoparticle? Is there any study on this?
Yes, sir. That is a very good question, sir. Because we have, why you want to use gold particles? Biocompatible one, mostly non-toxic. If you look at other particles like silver and copper, toxic and also unstable. So and also pH, if you change the pH, also nothing will happen. For gold particle, people are taking the golden particles for various applications. So nowadays working for respiratory disease also, people are conjugating with the gold nanoparticle. That is why gold is best one.
Yeah, in our traditional Indian medicine also they are
Yes,
traced
sir.
on sugar and bhasma and all these things.
Yes, I should also they concentrating gold particles, sir.
Yeah, yeah.
Yes, sir.
Thank you, sir.
Maybe any questions ask students to write to me also, sir. No problem, sir.
Fine, sir. Thank you, sir. Thank you
Okay.
very much, sir, for sharing your vast experience on nanoparticle, rapid detection of bacterial and viral infections to nanoparticles.
Because I started my career in maybe 1997, I started my career. Nanotechnology, India started very long back, very recent only started. I started maybe 10 years ago, advanced level. 1997 we started my work. My professor from Harvard University professor, he is passed out from the Dr. Whiteside, USA, Harvard University. He got some kind of information. He is a very strong background of chemistry and also synthetic person. He is doing a lot of wonders. Nowadays, he is working on the microelectronic industries, LED, solar cell. We can do a lot of things, but the students not interested for research. Simple technique, we can develop some methods. I got some many patents also recently, for 5-6 patents. And also I wrote some of the books also, silver nanoparticle I have book. I sent it to madam. Soon I can get some kind of, any student interested can come to my lab also, visit my lab. I can show you some demo also. We are always welcome, madam, sir. Madam, sorry, sorry. Okay.
Thank you very much, sir.
Yes, madam. Thank you, madam. I thank RItu madam only. RItu madam only is given the chance. I have to come to your place, madam. Sorry, because of COVID. Come to your lab.
No problem, sir. No problem, sir. We are very happy to hear you.
Yes, madam. Thank you, madam. Thank you. Any other questions, madam?
Yes, sir. We have covered a few questions. If they'll have any questions more, we can again contact through mail, sir.
Yes, madam. Yes, madam. Always welcome, madam. Thank you. Thank you very much.
Thank you very much, sir.
Thank you, madam. RItu madam, thank you.
Thank you, sir. Thank you very much.
We'll go ahead with the next session. In continuation of this GUJCOST sponsored national seminar, now I invite Dr. Shilpa Gupte to introduce our next eminent speaker, Professor Vandana Patrawale, Madam, Department of Pharmaceutical Science, Institute of Chemical Technology, Mumbai. Madam, please.
I, Dr. Shilpa Gupte, has a great privilege and honor to introduce our next speaker, Dr. Vandana Patrawale. She has completed her PhD and MPharm from the University of Mumbai. She is currently working as a professor of pharmaceuticals at the Institute of Chemical Technology, Mumbai. Her area of research include development of nanocarriers with a major emphasis on malaria, cancer, neurodegenerative disorders, medical device development, nanodiagnostic, nano vaccines, etc. She has more than 200 referred publications, 11 granted patents, 24 patents are in the pipeline, and 2 trademark registries. She has published 2 books and 25 book chapters with international publishers. She has her contribution, very active contribution in the teaching, research, and service throughout her career. She has awarded Kukreja Oration Award in 2020 and Dr. Manjushri Pal Best Pharmaceutical Scientist Award in 2019, Shri Amrut Modi Distinguished Research Award in 2018, then Woman Scientist Award in 2015, Bill Melinda Gates Grant Award in 2015, Best Pharmaceutical Scientist Award in 2014, then Vento Nanotechnology Award in 2013, APTI Best Teacher Award in 2012, and Fellowship of Maharashtra Academy of Science in 2011. So there are so many awards and recognitions she had received in past few years. She is Vice President of CRS Indian chapter. She is actively collaborating with the researchers as well as with the industries within the country and abroad. And she has completed Indo-Swiss, Indo-Japan, Indo-UK projects. She has executed all major grants from Indian government focusing on nanotechnology based product development. She has transferred many technologies to the industry and that include eluting stents, which is marketed in more than 60 countries. So with this brief introduction, I would like to once again thank to Madam who has accepted our invitation being a speaker of this particular GUJCO sponsored seminar. And we are really very lucky to have such a eminent scientist with us who will going to share her knowledge with us. So with this, I will like to invite Madam for her speech. Thank you, ma'am.
Thank you, Madam, for the kind words. And at the outset, of course, I would like to thank all the team members, the organizing team members of ARIBAS, in particular, of course, Dr. RItu Dixit for giving me this opportunity to be here with all of you. Can someone confirm if my slide is visible and
Yes,
I'm
madam.
audible, clear?
Yes, madam. Both are clear.
Thank you. Thank you so much. So what I'm going to do in probably next 45 to 50 minutes is talk about how we at ICT have been able to work on nanotechnology in healthcare, wherein specific focus is on translation. So whatever case studies today I'll be presenting, most of them have either gone to industry or are on way to be transferred to industry. So our focus is very simple. At ICT, which institute has given 19 Padma awardees to this nation, mandate is work on simple, scalable, cost-effective technologies so that they become really affordable. So obviously, we are integrating all the approaches in science and technology for sustainable future. I'm sure all of you know what nanosystems are. Anything which is less than 1 micron, that means less than 1000 nanometer, actually comes under nano. But if you look at US FDA, that's Food, Drug and Administration, they define true nano as something which is less than 100 nanometers. So keeping this in mind, depending on what application areas we are going to work in, we have to decide what is the size range which we should be going. For example, if you want something to be delivered to brain using the nasal mucosa as an entry, it has to be less than 50 nanometer. But if you want something which should go through your skin, the pore size is 200 to 300 nanometer. So you can go above 100 nanometer too. So it all depends on which is the site where you want to actually deliver the cargo. Now this cargo can be small drug, that is API, or it could be large moieties like genes, siRNA, certain proteins, peptides. So it depends on where do you want them to get delivered. Now this particular figure in a nutshell gives actually how these nanotechnology based drug delivery systems have evolved over a period of years. So you would see that if you look at 20 years back, majorly it were the lipidic systems. Even when I started doing my research in 1990, it was the focus of my research too. Then came nanocrystals, other kind of micellar systems, dendrimers, etc. And the past decade, that is past 10 years, have seen rise in nucleic acid therapies. And again, the lipidic systems came back. Reason being, if you have to deliver anything in the body, it has to be biocompatible. Not only biocompatible, it should be biodegradable too. And we all eat food. So obviously, food contain lipids, fats, oils, and they are getting digested by the body without any harm. So can you use the similar materials to fabricate nano carriers? Was a hypothesis which we framed, and we've been working in variety of areas. And you'll see some of the examples. And of course, if you look at last maybe 3 years or so, you'll see that even for vaccine delivery, the interest in lipid-based systems has increased. And you've seen now, mRNA vaccine, which came even for COVID treatment, basically prophylaxis, I would say, is lipid-based. So that says that this is one moiety which can be utilized in more than one ways to get the desired effect. The statistics, if you see, the business statistics, the commercial aspects, this is going to grow. This area is going to grow because if you look at cumulative annual growth rate, it is about 15%. The projected market is about 475 billion US dollars by 2027. So you can only just imagine how nanotechnology would come in our lives, day-to-day lives. Two mechanisms, basically, when you deliver drug delivery systems, how it reaches to the site of action. This is the cartoon which is trying to tell you that if nanoparticles are given by intravenous injection, when you have any kind of cancer in the body, there are leaky vessels which are produced. And you can see that they are so stacked here. But when you see near the tumor, there are leaky vessels. So the nanoparticles traverse through these leaky vasculature. So this is known as passive targeting, meaning you are not doing anything. You're just controlling the size, and by size, they'll be going to tumor. But if you want that it goes to tumor and doesn't go anywhere else, then you need to tag them with specific ligands which have affinity for the tumor. For example, if you are working on HER2 positive cancer, breast cancer, you have to tag with a moiety, maybe an antibody or an aptamer or a Fe body, which will take it to that tumor specifically and not go to any other organ. So by doing that, you're making sure that there are no side effects. What is the therapy? You know, when we talk about anything in healthcare, we want maximum effect, that means efficacy, and no side effect. Unfortunately, majority of the systems which are there in the market suffer from side effects because they are not targeted actively. Now coming to some of the studies which we have done at our end. So first study is where we are making nanostructured lipid carriers. And we have used it for combination therapy, dual drug therapy, specifically for cerebral malaria. Now you need to understand what are NLCs. Please again keep in mind, I've already told you our mandate is make simple systems. So what are we doing? We are just mixing solid lipid, liquid lipid, and surfactant. And when it is diluted in the body fluid, it will generate NLCs. So all you require is mixing vessel, a stirred vessel, no high pressure homogenization, no microfluidizer, nothing of that sort. So a very simple technology which has been given to industry. In cerebral malaria, actually what is happening is that once the parasite enters into the red blood cells, the vessels become hardened. So patient become comatose. Looking at the statistics of this malaria, 3.2 billion people, almost half the world's population, is at risk. And just as I am talking, with the next 10 seconds, one child dies of malaria. So that means that's the gravity, the situation. Having understood that, we need to do something for it. So what have we done? As I told you, we've understood pathophysiology of malaria very well. And that's where, when you're talking about biology and chemistry, coming the interface, no, talking about it, first thing is you understand pathophysiology of the disease condition, what happens, and then see whether you can give any solution by making nanoparticles or not. And a very simple solution which we have given is NLC. Next step is you've understood pathophysiology of malaria. You know how malaria parasite life cycle is. Next is which drugs to use. Go to WHO guidelines. Guidelines indicate what are the combinations which are preferred combination. When we looked at these combinations, we were surprised that for a condition like cerebral malaria, where hospitalization is must, fast treatment is a must, there was only one formulation available, and that also not a very stable formulation. 2 ampules or vials are given which need to be pre-mixed and administered. And it is a monotherapy, whereas WHO says it has to be dual drug therapy. Why not monotherapy? Because resistance get developed. So over a period of time, if resistance get developed, that therapy will not be useful in anyone. And when we look at the statistics, you'll be surprised that mortality rate, that means deaths from the severe malaria, approach 100%. So there is an unmet need, and therefore we formulated NLCs. When we looked at these NLCs, it has given 2 drugs which we have used, artemether and lumefantrine. One thing you have to keep in mind, that 2 drugs which you are using, one should be short half-life, one should be long half-life, and they should act by different mechanisms so that, you know, the malarial parasite is attacked from all sides. So having done in vitro characterization, including TEM, that's transmission electron microscopy, to prove that they are nano in size, we started with our animal experimentation. And in animal experimentation, you can see that when our formulation is given, even as monotherapy, initial work started with monotherapy. By that time, WHO came and said no monotherapy. So we tried to do dual drug therapy. But you can see when with monotherapy, parasite count is just 0, which doesn't happen with the marketed formulation. Then we went to malaria test. In that test, what is done is you are giving to the animals malarial parasite, and then you are starting the treatment. And you are counting parasites by taking smears from blood. You can see that the marketed formulation again has shown recrudescence. That means by the time treatment is given, it is okay. As soon as you actually stall the treatment, parasite count goes up. That doesn't happen with our formulation. And that doesn't happen even at 1/5, 1/20th, 1/50th of the doses, which is the therapeutic dose. That means you can reduce the dose manyfold. Having made sure about this, then we went to treatment model. In treatment model, what do you do? Just like in malaria, if a patient gets infected with parasite, you get fevers and chills. But they are after you get infected. So in this case, we allowed the parasites to grow and then gave the treatment. Unlike in the earlier case where we have simultaneously given. In this case also, you can see that our formulations are at base level, no parasitemia. Whereas with marketed formulation, again, you have seen as soon as the doses stop, the recrudescence is seen. We went one step ahead and then did studies in cerebral malaria. Now in cerebral malaria, it has to be done in C57 mice. They are black mice, special mice. So you need to generate cerebral malaria, and that you will know by measuring the rectal temperature of animals, plus their gait gets affected, they get paralyzed. So in this case also, our formulation has given us excellent results. So this particular patent is now a granted patent, and see such a broad patent. It's not only for malaria we have taken, we have said nano drug delivery based combination therapy for treating parasite infections. Because we know now if we'll use this particular system, it would be helpful to put any kind of drug in this particular platform. And we are actually testing it for many. Now, these are some slides which will make you believe that how good our systems are. So what have we done here? What you can see in red is rhodamine labeled NLCs, our nanocarriers. Wherever you see granular mass is RBCs where parasite has gone. Wherever you see blank, meaning they are normal RBCs. See, normal RBCs do not have any nucleus. So it's only when the parasite goes in, you will see such a nucleus. So when I start this video, you will see that very, very selectively, our NLCs are taken up by infected parasites. That means it is the chemistry of this particular system which is helping them go inside only infected RBCs. The normal ones remain as such. Further, when we did this testing in, you can see in C57 mice, this is after the treatment was given with marketed product. You can still see the gait of these animals. They are so paralyzed from one place to other place if they have to go, they take more than 15 minutes. Whereas you can see when our formulation is given, how agile they are. They become so normal, not paralyzed at all. So that means our system is able to do wonders and in fact give us efficacy which is at the subtherapeutic dose. Not only that, all these mice survived up to 55 days post-infection, which is their normal life. So veterinary surgeon told us, actually that you can use it for the next studies. Of course, we don't do that. We need to be very careful. So that is one study. So as was indicated, we are doing studies in infectious diseases, cancer, Alzheimer's. So I thought I'll take another study, which is from breast cancer. In breast cancer, if you look at the statistics, all women are at risk of cancer at all age groups actually, and statistics actually are mind-boggling. 1 among 8 women is at risk during the course of her life. And annually, more than 1 million women are diagnosed. So, meaning statistics were so bad that made us think, why is it that this happens? So, major reason, again, when we read about breast cancer, we came to know that major reason is when you have breast tumor, lymph angiogenesis takes place. That means lymphatic vessels grow. So as lymphatic vessels grow, what they do is they take the cancer cells and transport it to different organs. And if they transport it to different organs, all organs will start growing cancer cells. and that is the cause. So we thought, can we treat the sentinel lymph node? Sentinel lymph node is a lymph node which is adjacent to this tumor. If we can concentrate that particular node with lot of drug and do not allow it to go to blood circulation from where it is going to different organs, we'll be able to kill most of the cells. So this slide just tells you that yes, if you are having METs, that means the metastatic cancer cells going into different organs, it's going to affect all the organs. So that will lead to decreased patient survival. So our hypothesis then was, again, to make analyses. As I told you, just mix ingredients, but you need to select ingredients very, very critically. These ingredients need to be GRAS approved or US FDA approved. They have to be used in ratios in which they are safe. So our hypothesis is, we'll give them by oral route, let them go to the intestine. From the intestine, just as lipid metabolism takes place through chylomicrons, they'll be taken up and they'll go to lymph first, partition in blood later. So now if they are going to go to lymph first, that means they'll go to sentinel lymph node in higher amount. So to prove this hypothesis, we took the gold standard drug which is in use in market, tamoxifen citrate. Now this particular drug, as you can see on the slide, has n number of problems which need to be addressed. Both the pharmacodynamic and kinetic associated, as well as toxic effect to each and every organ, which is not good. Patient may recover of breast tumor, but what if he or she, in this case only she, will have all other problems? So what have we done? We have formulated NLCs, and then we started with in vitro cell line studies. We typically do cell line studies in 2 cell lines to confirm, reconfirm. So MCF7 and ZR75-1 are the breast cancer cell lines, and you can see that our NLCs are given results equivalent to that of tamoxifen when it is taken in solution form. Now keep in mind, tamoxifen is not a soluble drug. So in this case, we have used organic solvent to dissolve it. What are we trying to interpret from these results? We are saying that NLCs are behaving as good as the drug in molecular form. So once we established that on both the cell lines, we went ahead and looked at cell uptake experiments by confocal as well as by flow cytometry. And we saw that they were getting accumulated in cytoplasm, perinuclear and nuclear spaces of cancer cells. And this was done by even Z-stack. Many times 2-dimensional confocal can really give you ambiguous results. So by even the Z-stacking, which we proved that it is a nuclear co-localization which is happening. Next was that we had to move to preclinical studies. So when we moved to preclinical, when the student looked at this data, he was little dejected, because this is what marketed gives, faster onset. This is what our formulation is. But I was happy because it has proven our hypothesis that the formulation is going to lymph first and then it starts appearing in plasma. These are the results, pharmacokinetic results in plasma, right? So typically, that is what has to be taught to students, that observation is what matters. So don't see that you want same profile as marketed. If you're getting something different, there may be something good in it. So interpret that. So we have seen here that Cmax is, of course, 2.714. T half is 7.10, which is really, really great. That means you can keep the drug in its effective concentration for prolonged period of time. This is biological half-life. You are extending that. From 1 hour, you are able to extend it to 8 hours. That means the frequency of administration will decrease. So that is very important. Now, this was a study which gave us enough confidence. Marketed is not taken up at all by lymph, whereas our formulation is taken by lymph. And this really proves that first it goes to lymph and then it comes to blood. And this also proves that if it does that, that means there won't be any side effects. So when we plot this, you can see that marketed sample 3 mg per kg after 28 days, it is reducing tumor volume, but not as effective as our formulation. It's shown here also. You can see marketed. It's almost same as, in fact, little bit better than untreated. But see what happens at our formulation. Even half the dose is much better than the marketed. Now, in cancer, what is important is there shouldn't be, as I said, side effects. That means patients should survive. Tumor can be controlled, but does patient survive? So in this case, you can see that 50% mice, actually they die. Whereas in our case, 100% survival was seen. Again, saying that lipidic formulations are safe, and this particular formulation, since it's taken by lymphatic route, it is really very safe. We again coupled it with enzyme test, and you can see marketed, all enzymes are going haywire. They are increasing, SGPT increasing, SGOT increasing, alkaline phosphatase is increasing, and of course, lipid peroxidation is happening. The aldehyde is getting formed. Whereas with our formulation, they are as good as normal. So we proved that nothing happens to the organs even by histopath, and therefore concluded that we could actually make these nanoparticles. Another case study which I'm taking is cationic system. Now, why cationic? Because we believe when you have a cationic lipid, which we in-house synthesized, I'm just giving you a hypothesis so that you are free to think, what can you synthesize? Chemistry students can think, how can you synthesize? Biotech students can think, how can they make use of these and do cell line testings? And pharma students can definitely think how they can make drug delivery system. So luckily in our lab, we have biotechnologists, we have pharmacologists, we have chemists, all of them. So we are able to do this kind of work. So hypothesis is when you have a cationic lipid, it is going to interact with lipid bilayers. Now lipid bilayers are present in all membranes in your body. So when it interacts, how the reversal happens, you will see. There's a flip-flop mechanism. That means anionic endosomal membrane lipid, which you are seeing here, it is going to get flopped. So when it flip-flops, there is a stable cone complex which is formed, as you can see in this slide. And because of which, there would be reversion to inverted hexagonal structure. And this is what is required. Why it is required? Because your moiety will be taken up by membrane. It will go inside cytoplasm, but it has to escape endosome. In order to release the cargo near nucleus, it has to change in its structure. So we had this hypothesis and we started working with this hypothesis. So now we have range of cationic heterolipids, but and this one definitely is patented, and I'm not able to give details of the lipid. All I can say is it is a heterolipid. Hetero meaning it will contain heteroatoms, and it is cationic. That means it will be positively charged. And positive charge we have given using guanidine group. So it is guanidino heterolipid. That much I can tell you. But the chain length, etc., sorry, I'm not able to reveal because the technology has been given. So we formulated SMED systems here, both cationic and non-cationic. What are SMEDs? They are self-microemulsifying drug delivery systems. NLCs contain solid lipids. SMEDs do not contain solid lipids. So what do they have? They have only lipid, surfactant, and co-surfactant. That's it. When they go in the body, they will make a microemulsion, which is nano-sized. And you can see the size here. It is less than 50 nanometer. So both cationic and non-cationic are giving less than 50 nanometer. Only difference is the size. Okay, only difference is the charge. So you can see the cationic one are giving us positive charge. So in this case, the study was on melanoma. Etoposide was a moiety. So you can see the cell line studies which have shown that complete morphological alterations happen. The clonogenic assay is where you are giving treatment with your preparation. So if you have cells growing and you give treatment, ideally they should all die, just as I've shown in the case of ECS. ECS is etoposide cationic SMID. Nothing comes back. But if your formulation is not very effective, you'll see the cells growing after 3-4 days. So this is non-cationic and this is cationic. So we went further and did cell cycle analysis, and very clearly you can see in the cell cycle analysis that when we are using ECS, there is 83.92% cell arrest. Of course, in the G22M phase, because etoposide acts in G22M phase, but highest by this particular formulation. Then we looked at what could be the mechanism of uptake. For that, you need to give different treatments. For example, if you're wanting to see whether it is energy dependent, you give treatment with sodium azide. And if there's no uptake, you know that this is the mechanism which is more predominant. You can treat the cells at 4 degrees Celsius and then do experiments. All these are fluorescent labeled SMEDs, which we have used. Or you can treat it with colchicine and chlordiazepoxide to know whether it is any other pathway. So when we did this study, we could make sure that ECS, that is cationic SMEDs, have possibly followed clathrin-mediated pathway. Cytoskeletal is when you give colchicine, and it's also partially going through energy dependent. So it's more than 2-3 mechanisms which are making it go towards the nucleus. Look at these results, excellent results. Marketed, hardly any efficacy. And ECS, your cationic SMEDs, even at half the dose, again, is much, much, much better than marketed. P less than 0.001. What more do you want? So when this happened, again, we looked at the survival. Survival is excellent with the ECS group. Morphological alterations. See, when marketed is given, you do see polychromatic, hypochromic, anisocytosis, cytoplasmic vacuolation, all changes in blood cells. Nothing happens when our formulation is given. And all these studies are not done at our end, so there's no bias. We generally do it from certified labs, and our collaborators are ECTRIC and NIRRH. So absolute great results. Now this same thing, the lipid, we've also used it for gene delivery. In this case, what did we do? We have made monogwenoplexes. Again, just mixing. So you have solid lipid, surfactant, and to it, small amount of cationic lipid is put. So it's only variation of these systems, which we are attempting. And then if you incubate it with any kind of negatively charged material, DNA is negatively charged, siRNA is negatively charged. So it could be any gene. So we have now worked on breast cancer, prostate cancer, lung cancer, many others. But I'm just giving this representative. Main thing, before you start your work, you have to make sure that your moiety has got so very attached to your system or is encapsulated in the system that when you treat it with DNAs, it doesn't break it down. So you can see that if you take naked DNA, in presence of DNAs, of course, you know what is going to happen. It is going to eat it up, right? If DNAs is not there, you see all the bands. Same thing with our monogwenoplexes. If DNAs is not there, you'll see all the bands. But here, in presence of DNAs also, you are seeing all the bands, making sure that yes, your moiety is stable. It has conferred protection to the plasmic DNA. And then we did anti-proliferative studies, and you can see that lipofectamine is known as a gold standard. Disadvantage of lipofectamine is it's very toxic, so it cannot be given internally. Now we have a system which is almost as effective as lipofectamine, but not toxic at all. We have completed acute, chronic, carcinogenicity studies, genotoxicity studies, mutagenicity. All the studies are completed, and you can see the transfection of monogwenoplex is as same as lipofectamine. So obviously with this, we had the confidence that yes, we have a system which can definitely be used for gene delivery. And this also has been given to a company. Now, where are we moving? Definitely, we are looking at modifications of the nanoparticles, either by antibodies or nanobodies, which are smaller antibodies, stable ones. We are delivering nucleic acid. We are using cell membranes, extracellular vesicles as future, of course, is for CAR T therapy. I'm sure all of you would have heard about this chimeric antigen receptor T therapy, where T lymphocytes from patients are taken, right? They are treated, you're growing, you're changing the defect, whatever is there. So editing the genes, putting it back in the body. So CRISPR and CAR T is something which is coming up. So slowly we are moving in that. How much time do I have? If you could tell me, I'll accordingly go ahead.
Yes, madam. Still, we can continue for 15-20 minutes.
Oh, that's great. Okay. So this is another study which we did. Now I've given you the therapy part. I thought let me also give you some diagnosis where we've been working, and excellent lecture by the previous speaker. But what you will see in my, this is a case study, the actual case study. So what have we done here is we are working on veterinary, basically infections, and brucellosis is one such infection which is caused by B. abortus. And if it not only affects actually animals, it also affects human beings. So if you are eating dairy products, you can get brucellosis also. If you are farmers, who are in very close vicinity with these animals who are infected, will also get this. So what have we done here is we looked at whether we can come up with point of care testing. In earlier presentation, you saw they are doing a lot of work with serum. But the question which I ask student is, I want to avoid serum, I want to avoid plasma. Can this test be done from saliva? Can it be done from milk? Can it be done from urine in a sensitive way? So we did a lot of literature survey and then came up with that we'll use nanosensor. Now what are our nanosensor? He talked about gold. We have silica nanosensors. And now we have a very broad patent again on silica nanosensor. Now you may ask, why silica? Cheapest material, comes from waste material. Anything which is burnt, any crop which is burnt, silica is generated. So dirt cheap. So we thought we'll make fluorescent silica nanosensors and use it for making a kit for brucellosis. So how did we go? As I told you, we made silica nanosensors, and this silica nanosensor we have actually published. You can see the reference which is given at the end of this particular slide. After making nanosensors, which is by chemical method, we adsorbed our antigen in it. And this was actually coated onto the surface as he indicated. So it's a lateral flow assay kit. It has to be assembled. So there's a plastic case over which there's a nitrocellulose membrane which runs through. Then you have to have a sample pad. You have to have an absorbent pad which can absorb any excess material. You have to have the control line and a test line. So you are going to put your antigen on this if you are detecting antibodies, or you will put antibodies on it if you want to detect antigen. It's as simple as this. So all you have to do is, once you are entering into field, we were very new to this field, maybe 7 years back. But now you ask me, I have n number of kits ready and commercialized. So you need to think, you need to read, need to understand, and then come out with cheap, simple, efficacious methodologies. So what did we do here? Is, yes, as I told you, we have taken our own FSNs, that's fluorescent silica nanosensors, which we characterized in detail. And then we have used it for detection of antibodies. Now we have done it in milk, we have done it in urine, we have also, just to start with, we have also done it in blood. I have to see whether it works or not. It shouldn't happen that it doesn't work in milk and therefore we say it doesn't work at all. So we've also done in, of course, sedum. Now you can see what this slide represents is all the different tests which are used currently for brucella. PCR is most expensive and sensitive of all. ICDT is what we have done. It stands for immunochromatographic diagnostic test, and it has ICT in it, so I thought I'll put it as ICDT. So we've compared it with PCR, serum, or milk. We've compared it with you routinely use Rose Bengal test or STAT test, ELISA also, and you can see that ICDT with respect to sensitivity, specificity, it is in agreement with ELISA and PCR. Having done that, of course, again, it's a granted patent, and plus this technology was awarded by President of India, and the student is, we've sold the technology actually. Ideally, he should have become an entrepreneur, but the interests were different, so we've sold the technology. Now, this slide indicates, what TRL you are. Generally, academicians feel that they are at TLR of 8, 9. Not so. If you have proof of concept, you have only reached technology readiness level of 4. After that, you have to do lot of validation, lot of GMP manufacturing, lot of testing to reach TLR 9. We are also working on stents. Stents are used as actually alternate to bypass surgery. And now we have started actually coating nanoparticles on devices. In fact, I'm working very closely with this Gujarat-based company, let me tell you, who are market leaders now. In fact, we were the first one to get even the CE mark. So you will see this video, which will actually clearly tell you how we have gone about. So when there is a plaque in the vessel, you are actually reaching it by this guiding catheter. Onto this guiding catheter which goes, you have balloon. And on the balloon, you have this stent, which is spring-like structure, which is crimped. Now, the balloon which is there has to be really very thin balloon. It should inflate, when you're pumping, it should inflate. When it inflates, obviously the stent will expand. Now, in addition to that, this particular technology requires that you have radiopaque markers, because they are the ones who will decide where the plaque is and where the interventional cardiologist has to stop when they are taking it through your thigh muscle or shoulder muscle. So you can see here now that it is going through the blood vessels in, balloon is wrapped onto it. It has to have a shape folding memory and very, very low crimp the stent should have on the balloon. Otherwise, balloon cannot be taken back. That would really create a chaos. Many times such failures have happened. Now you can see this orange color plaque. This is a plaque which gets deposited when you are eating high fat food. Okay, majorly all your fast food is giving these problems. So you can see as soon as this is located, interventional cardiologists will go on expanding. So when you are expanding the balloon, the stent is getting expanded. You have to go on expanding so that there's longitudinal expansion and radial expansion. Now it is compressing the plaque against the artery. It doesn't push it further. It just compresses. And once that happens, and you're making sure that the stent is locked in place, after locking it in place, you would take out the balloon. So once the balloon is taken out of the vessel, only stent will remain inside the artery. So this is the mechanism, what happens, okay, as far as stent is done. Can I tell people to please go on mute? It's really disturbing.
I request a participant, please mute your mic.
So you can see
Miss
now that,
Mithal. Yes.
The catheter is taken
Miss
out
Mittal.
and stent remains in place. So what have we done is that we have actually coated onto these stents our drug as nanospheres. We have evaluated that in pigs, and all these studies are done in Netherlands. And what is important is there should not be any late loss. It should be near zero. That means when you planted stent, you opened up the vessel. It shouldn't happen that again the stent actually shrinks. So that shouldn't happen. So not only that, we've done studies in human beings and we've shown that our drug-eluting stents are much better than bare metallic stents with respect to all the parameters. We've also shown by doing optical coherence tomography studies, which are as given in the slide, that the lumen is opened and it's as good as what market leader Abbott stent gives. In fact, this was in papers all over and the company has really got many awards. So of course, we started with first stent, which was paclitaxel, went on to sirolimus, went on to everolimus, and now we are going to many devices. We have made changes from stainless steel to cobalt chromium to nitinol, so on and so forth. So many of these have been actually marketed, and as Dr. Sheetal introduced, 65 countries these are being placed, but there are still challenges which we are trying to tackle. This is some work which we have done during COVID period. And why I'm showing here is that you can do really simple suspensions in market for these foals. Foals are, babies of horse. So typically they die if they get diarrhea. They eat something which is not, adjusting with them. Affects more than 20% foals. Right now, antibiotics were given. So we thought, can we come up with something where no antibiotics are given? But they are able to take care of dehydration, take care of acid-base imbalance, no loss of electrolytes, and without disturbing microbiota. Antibiotics will do that, right? They'll disturb microbiota. So here, we've come up with the formula. What we did is, Mumbai was worst affected by COVID. So I identified a student in Ireland who had graduated from ICT and was doing masters. He was about to finish his masters. Told him to go to this company, and daily, night, I used to give instructions and morning he used to work. And within 3 months, we came up with this product, 2,4, okay, which is non-antibiotic. So it's prebiotic based. And why nano? Because we are using clays here. So we have actually very fine clay used there, which is given us the best results. And another last study is through Startup India, wherein what I'm trying to show with these two examples is simple things can work wonders only if you identify the unmet need. Now, these dental alginates, which are used to take prints of your tooth, when you go to dentist, were all imported. Not a single product was existing in Indian market made by India. So what we did, now this is marketed product through transfer of technology from our end. Marketed product was also in these pouches, big 1 kg, 2 kg pouches. We thought we'll give to the dentist sachets so that when this is opened, rest of the material should not absorb moisture. It is not wasted. So we did market research, did surveys, identified that dentists would like to have this thing. Another thing which we did is we put 2 indicators in it. So when dentists are mixing, they were looking at their watch continuously, We said when it changes the color, you stop. Then put it on your teeth surface, again it will change color. It has to be taken out and put into a media. Also, kids used to throw it out. They used to warm it. So we also added flavors to it, strawberry and chocolate flavor. Any flavor can be added, but this was the, when we did survey, this is what was preferred by moms and kids. So we went ahead with this. So this product is also in the market. So all these studies, translational kind, were possible only due to support, which we got from government, through various academic institutes, our collaborators, and of course, a lot of industries. And this is our lab. We have all the facilities in-house for doing any kind of nano work, any kind of biotech work. And this is our happy family. And they really work as a team to see that any product which we take from industry goes to the level of translation. So with that, I would stop and take questions, if any, from the audience.
Thank you very much, madam, for sharing your vast experience with us within such a short time. It was indeed a fruitful session on use of nanotechnology in health science. Now session is open for discussion. Any question from audience? We have already taken few questions raised. May I, madam? May I ask few
Please.
questions? We have collected few questions.
Please go.
Madam, you developed a kit for the diagnosis of brucellosis, which is comparable with milk and serum PCR, ELISA, and RBT. What is the exact sensitivity and specificity?
So we can go actually in the nanomolar level. And as far as specificity, I told you it is 100%. No, what ELISA is giving, PCR is giving, same as our kit giving. So obviously it is absolutely on par with them. Highly specific. We've tested different organisms also.
Okay.
Because this is an antigen antibody reaction, please keep in mind. So it is very, very specific to that particular organism.
Yes, based on affinity, right? Next question is, madam, one student is asking, what is this fluorescent silica nano sensor? If we can just brief this.
Okay, so as I said, no, silica is a very cheap material which comes from any of the crops when you burn. So silica is also being sold in the market. So what we have done is we have tagged it to a fluorescent sensor. So you can take, say, for example, fluorescein as a dye. No, fluorescein is a green dye, right? So we have loaded fluorescein onto these silicas and made it in such a way, it's a chemical reaction, so made it in such a way that it gives us nano size. It is well, in fact, as I told you, no, you can go through the basic paper to get an idea, but essentially you are reducing it while adding the dye, and then you're making it free of any free dye. So what is very important is that you have the material which is within and which is on the surface of silica. You shouldn't have any free dye. Otherwise, it will interfere with your test. So you dialyze it out and then purify it and then use the material. So as a name itself suggests, it's fluorescent tagged silica
nano
sensor.
Correct. Thank you, madam. Next question is, can we design nano-based sensor?
Yeah, so I think this is one. So you've seen in the earlier presentation also, people have tried gold nano sensor, right? People have tried, he also said people have tried platinum. There was a question, people are trying carbon nanotube now, they are functionalizing it. So any different types are there, and latest of course is quantum dots. We are also working in quantum dots, but our focus is on herbal. Can we make quantum dots using natural moieties?
Correct. Okay. Next question is there any medicine based on lipid? Or we can say
there are many. In fact, we have marketed. So we started with, again, with simple thing like curcumin. So it is now being given to all the hospitals through Cadilite is being marketed. So that's our technology, which is gone. And another one is where we have, you would have, in the COVID period, people would have taken vitamin D capsules. So vitamin D in the soft gelatin capsules, again, lipid-based technology, which we have worked together with them. So, and maybe probably the second slide or so, I showed many drugs which have gone to market, including amphotericin, which was in shortage, right? Amphotericin liposomes were in shortage during COVID period.
Correct.
So, which is used for systemic fungal infection. Again, that is one which is in the market. There are many, including doxorubicin, paclitaxel, many.
Fine. Thank you, madam. I think Dr. Hemant sir wants to interact. Yes, sir.
Yes, good afternoon, madam.
Good afternoon, sir.
Very nice presentation.
Thank you. Thank
I
you
have
so much.
read somewhere the vaccine, nowadays vaccine technology is based on these lipid vesicles.
Yes, very, very correct.
mRNA based techniques. So how they have done this and make the formulation?
So what we have also seen, the malaria, what I had shown, NLCs, right? Those NLCs, if you look at, they act as adjuvants also. So they are making lipid nanoparticles like NLCs and loading mRNA into those. So again, this particular part Serum Institute wanted from our side, because we have synthesized many. So if you have a cationic lipid, as I told you, it will interact with any kind of negatively charged materials. So when mRNA is negatively charged, so you have a ready base, and then you can encapsulate any kind of cargo in it. So mRNA, yes, very correctly said, mRNA vaccine, which is there in the market, by Moderna, by Pfizer, all are lipid based. Yeah.
Thank you.
Fine. Thank you very much, madam. Is there any other question from audience?
In fact, for COVID, we have got DBT Direct grant, and we are also working on, but we are looking at nasal route. I don't believe in pricking everyone. People have to go to the hospitals or to the caretaker. So we are developing a nasal vaccine, and studies are ongoing in animals. So again, for COVID. Yeah.
Fine. So thank you very much, madam, for sharing your knowledge and expertise with us.
My pleasure.
I hope this question answer session has cleared majority of the queries, and we can continue with this next session. Thank you. Thank you very much, madam.
Sure. Thank you so much. Bye.
Bye. Okay, ma'am. Have a nice day, ma'am. There are some instructions for the participants who have participated in oral and poster presentation commencing at 1 p.m. It is our humble request to those participants to join their respective rooms through their corresponding links before 10 minutes. Hope everyone has received the links. Now we'll continue with our next expert talk. I invite Dr. Jinabai Chauhan to introduce next eminent speaker, Dr. Hemant Soni, Associate Professor, Department of Chemistry, MS University, Baroda. Dr. Jinabai Chauhan, sir, please.
Good afternoon to one and all. It is a great pleasure for me to introduce third speaker of today's seminar, Dr. Hemant Soni sir, who is going to talk us related to nanotechnology. Sir is currently working as an associate professor at Department of Chemistry, Faculty of Science, MS University of Baroda. Sir obtained his master and PhD in applied chemistry. Sir is having 16 years of academic and research experience. If I highlight research expertise of Dr. Soni, then Sir published 26 research paper in various international journal. If the total citation we can see, then it is 552. The H index is 13. 2 students awarded PhD under the guidance of Dr. Soni. 8 students are currently working. Apart from this, 1 postdoctoral fellow is also working under him. The number of projects assigned, completed or ongoing, that is 4. And sir has delivered several lectures and talk related to nanoscience, nanomaterial, polymers, drug and gene delivery materials, photocatalysis, organocatalysis, contrast agent for magnetic resonance imaging in a various scientific gathering. Sir has been awarded with Dr. A.K. Shah Best Research Paper Award in Chemical Science by Gujarat Science Academy in year 2019. Sir is also serving as a member of Board of Studies in MS University and other university. Sir is also associated with Kendriya Vidyalaya of Gujarat region. So with this brief introduction, I welcoming Dr. Hemant Soni sir for his talk. Dr. Hemant Soni sir, please.
Thank you, doctor. Just I am presenting my screen with this. Okay, this one, share. Is this visible?
Yes, sir. It is visible, sir.
Just wait. Yes. Now, I want to make a PowerPoint presentation of this. Now the screen is visible, madam?
Yes, sir. It is visible.
Okay. So
We can
first
make
of
it as,
all,
we can, sir,
yeah,
make it as a full screen.
yeah. Now is it okay?
Yes, sir. It is visible.
So first of all, I am thankful to the organizers of this event and congratulate them for arranging such a nice event. So in first two lectures, we have seen lot of things and what of the possibility with the help of nanotechnology, what can we do in the area of biology, chemistry, or even physics. So today I am going to present the combination of all this branch in the form of developing the iron oxide nanoparticle based on drug delivery platforms and T1 T2 dual contrast agent for magnetic resonance imaging. For this purpose, we have to first understand some chemistry, some biology, and some physics also. So slowly and slowly, within a 45 minutes, I am going to explain you what is it and how we work in this area. So in a brief, this is the one of our works carried out in my laboratory. I am going to present first, and then I will show you how the development was going on. So we synthesize Fe3O4 nanoparticles. It is called magnetite nanoparticles, and cover these nanoparticles, encapsulate this nanoparticle with EDTA. And then hold this assembly is again encapsulated is polymeric micelles called pluronic F127s. Now whole this assembly was used as a carrier to entrap the drug for drug delivery purpose and also used as a contrast agent in MRI. We will also publish this paper in one of the best international journal. So what is the use? Why? See, in the first lecture, we have seen about the gold nanoparticles. In second lectures, we have seen about all these lipid-based carriers. Now, in third, these lectures, I am going to show you how magnetite nanoparticles, which is magnetic in nature, can be useful for the same purpose. The best way of using this nanoparticle is to first we can anchor anything on it. Whatever the delivery cargo, whatever the requirement, accordingly we can modify it, we can make a shape of it, and then we can attach the targeting agent, gene therapy agent, therapeutic protein, chemotherapy, drug, magnetic, and this whole assembly can be used for the magnetic hyperthermia purpose, which require one more lecture. But I am not going to shift in this magnetic hyperthermia, but I am going to show you how this drug and gene can be loaded on this pion. What is pion? I am going to show you. So this is all about the architecture of nanotherapeutics pions. Okay. So nowadays, nanoscience, nanotechnology are boon to the patient suffering from the diseases like cancer, diabetes, Alzheimer's. Okay, yes. So especially in the case of cancer, as we have seen in the first two lectures, it requires early and explicit diagnosis followed by the target specific treatment. First of all, we should know about the from the very first stage of the cancers. Once we know cancer is developing, then the treatment is required. The traditional treatment like chemotherapy, we know this is a vigorous and non-specific. Ideally, factors like psychology of the patient, efficacy of the drugs for the disease, immune response, competence of the patient towards the drug doses should be considered during this chemotherapy. and it should be considered. And the psychology of the patient is the main thing. It should be considered before adopting any treatment for the cancer. We know which drugs are established for this. Cisplatin, Taxol, Paclitaxel, doxorubicins are the available drugs in the market. As Madam explained, these drugs have many side effects in patient due to non-specificity and insolubility in the body fluid. We know about this in the previous lecture. Now, as we have seen, many strategies have been developed for the purpose. For example, drug is entrapped in micro or nano cavity of the carrier, another carrier. We have seen the lipid as a carrier. We have seen gold nanoparticles. Some other carriers are micelles can also be used. Cyclohextrin cavity can also be used. We have seen liposomes, lipid bilayer sheets can also be used for the purpose. So these are all having, we have to first manage the hydrophilic, hydrophobic surface or balance is required to entrap any cargo in this carrier. This suffers from the disadvantage of enzymatic degradation. What is our purpose? We want to bring this cargo, allow this cargo to transpect the cancerous cell. Once they transpect or enters the cancerous cell, they should degrade and release the cargo. So in between their path, from the beginning to the cell, target cell, there should not be any degradation. But our body has developed, through the process of evolution, our body has developed some degradation mechanisms. They consider this cargo, they consider this carrier as a foreign body. And as they consider this carrier as a foreign body, they try to degrade it. So first of all, some enzymes are released to degrade these carriers. So we have, if we want to bring this cargo, bring this carrier towards the cell, towards the cancerous cells.
Excuse me, sir.
First, yes, madam.
Sorry, sir, for interruption. Sir, your slide is not moving. I think this is the first slide only what we are observing.
Okay, sorry, madam. Yeah, just wait.
Yes, now it has been changed.
Now, is it changed?
Yes, sir, it is changed, but full screen nahi hai abhi bhi.
Now it is changed.
It is changed.
Can you show change?
Yes, sir.
This one.
No,
okay.
sir. No, sir. Now it's not changing.
First slide. Can you see first slide?
Now it is second slide, sir.
Second slide. Now third slide.
No, sir. Only second slide. It is not changed. Yes, sir. Third slide.
Third slide. Then fourth. Now can you see?
No, sir.
This
This
is fourth
third
one.
only. No, now it is changed. Yes, sir. It is fourth one. Yes, sir.
Fourth one. Fifth one.
Yes, sir.
Sixth one. So I think I have
Yes,
to
sir.
remain with this mode only. Is it
Yes,
okay if
it's
I am
fine.
remaining
Yes,
with
sir.
this mode?
It is visible. No problem, sir.
Okay. So just again to revise this. So what we have done, madam, can you see second slide? This is second slide. Hello?
Yes, sir. Second slide is visible.
Okay. Now, this is second slide. What we have done, we have developed epithelial nanoparticles. We have encapsulated this with EDTA. Then we encapsulated with the polymeric micelles, and we use whole this assembly for the drug as a carrier for drug delivery purpose and for MRI contrast agents. What is it? What is the use of? See, in the first lecture we have seen gold nanoparticles. Second lecture we have seen lipid nanoparticles and the silica as a nanoparticles. In this lecture I am going to show you how magnetic nanoparticle like magnetite can be used as a drug delivery platforms. Okay, we have used this anti-cancer drug for the purpose. Available drugs are there, but these drugs are loaded with many side effects. To decrease the side effect of these drugs, target specific treatment is required. Okay, so what we have done, what are the available techniques? Lipids are there, we have to entrap the lipids, we have to enter the drugs inside the lipids, but we have to take care that these carriers should not be degraded in between during its journey towards the cancerous cell. Okay, even if these drug carrier is successful to reach towards the cell, cell transfection is the major issue. In the previous lecture, we have seen that Madam has used that charge electrostatic interactions for the cell transfection purpose. But every time this electrostatic interactions is not feasible, we have to take care of other techniques for the transfection is required. For example, we have to attach some adapter, some recognizing elements called folic acid or folate receptors onto the surface of these nanoparticles, which can detect the cancerous cells and transfect inside it. Madam, is it visible? I am on the 7th slide. Is it visible? Yes. Hello, am I audible? Hello.
Yes, sir, it's visible. You can carry on.
Yeah, thank you. Okay, so I am finding the gap. What was missing in the previous two lectures? See, so what were the major physiological barriers encountered by a typical therapeutic vessels? First of all, blood itself is a great barrier for this carrier to reach toward the target cells. So how bloods encounter this? Blood is a highly complex fluid. The enzymes are released in the blood. This enzyme degrade this carrier. Even immune system can recognize this carrier and degrade it. Even if it is successful to going through the blood, another hurdle is the liver and spleen. We know this object larger than 100 nanometer recognized by the liver and spleen and removed from the circulation system. So what is the solution of this? We should have the nanoparticles or carrier having a size less than 100 nanometers. How can we excavate the blood? If we are pagylation, if we are doing the pagylation, what is the pagylation? To cover this our carrier with the polyethylene glycol. If we have a deuterionic polymers, then it can escape through the blood. Liver, to escape through the livers, we should have a hydrodynamic size less than 100 nanometers. Another hurdle is the kidney. If any object having a hydrodynamic size around 100 nanometers, they are filtered by the kidneys. Object other than 10 nanometer pores of glomerulus are filtered out from the bloods by the kidneys. So we have to take care about the size of the nanoparticles. How we can do this? I will show you. Another hurdle is the blood-brain barrier. Tight junction between the endothelial cells in the brain prevent the passive access. There is a very tight packing between these endothelial cells, as madam is shown by the video. But in cancerous cell, there are some leaky pores. Through these leaky pores, we are successful to transfect or to enter this material carrier into the cell. So gas between the endothelial cell restrict the material escape from the blood. So it is called EPR effect, enhanced permeability and retention effect. Because of this EPR effect, what is the meaning of EPR effect? There is a leaky pores in cancerous cell. So endothelial cell lines are not very tight in cancerous cells. So we are able to transfect our carriers through these cells into the through this cell wall into our targets. So we can take advantage of this effect. So hydrodynamic size of 30 to 100 nanometers, or it is observed that even the cargo having 400 nanometer hydrodynamic size can be entered through the cancerous cells through this endothelial cells of cancerous cells. Okay, cell line of the cancerous cells. Now let us see. So how can we do all this job? Surface engineered super paramagnetic iron oxide nanoparticle. What is the full form of spions? Surface engineered super paramagnetic iron oxide nanoparticle are the best option available for the purpose. The advantage of these spions are, as a drug carrier are, we can, over the traditional drug delivery vehicles are, we can tune the particle size. We can tune the shape and surface charges according to our requirement. So how this parameter affect the efficiency of the delivery vehicles? For example, shape affect the biodistributions. Spherical shape is the most affordable one, most effective one. Its shape affects the cell toxicity also. Size of the particle, as we have seen, liver is there, spleen is there, kidney is there. So to escape through these hurdles, the size matters. And this size determines the residual time in the blood. We require longer residual time of our system inside the blood. And its size also affects the magnetic properties. We require magnetic property to act as a contrast agent in MRI. We know this MRI machine contains a magnet, and magnetic system directly affect the imaging of the object. I will show you. Now, surface property also affect this efficiency of the material. Surface property indicates the colloidal stability. It determines the internalization into the target cells. It was observed that particles with a size larger than 200 nanometer are removed from the body by liver and spleen and reticular endothelial system. You know this as a biology background. Less than 5 nanometers are rapidly excreted to the kidneys. I have shown you. However, the circulation lifetime of the particles in the blood can be extended and tuned by varying the nature, which is called hydrophobic, hydrophilic nature of the coating material around the core. I am going to encapsulate my carrier with the coating materials, and which have a HLV value, balanced HLV value, depend upon my requirement. Okay. Now see this. So magnetic nanoparticles have a nature. Now coming towards the magnetic nanoparticles. When I have any magnetic materials, I am going to the nano region, this particle have very, very high surface area. Due to the very high surface area and magnetic property, this particle have natural tendency to agglomerate, to coalesce and make a larger structures. And if larger structure is formed, their size increases. And if their size increases, our circulation, that hurdle, that system, that liver and kidney easily recognize them. Enzyme can easily work on them and degrade them, which is not desired. Which is not desired. So we have to control over the particle size, and we have to prevent this magnetic nanoparticle to agglomerate. We have to prevent the agglomeration of these particles. Now, how can we do this? Now come to the technical terms. What are the technical terms? We required a nanoparticles which are magnetized only on exposure to the applied fields. See, they are coalesced, they have a tendency to agglomerate, but when they are magnetic in nature, so what I will do? I will have a particles in nano size, but they are non-magnet, and they become magnetized whenever required. In presence of magnetic field of MRI, in presence of magnetic field of MRI. So when I keep my lesion, my object under the MRI, then and then these magnetic particles become magnetized. Otherwise, under normal conditions, they will remain non-magnetized. This type of property of magnet is called superparamagnetism. Okay, so these particles should be immediately demagnetized on removal of the field. And because of this, I can prevent the agglomeration of the materials. So in technical terms, it can be called the material should have zero coercivity with no hysteresis. Zero coercivity means there should be not any magnetisms, and there should not be any residual magnetisms. That means you may observe that in student point of view, if I am explaining, you may observe normal magnet. When I keep any iron material in contact with the magnet, and when I remove the magnet, that iron nail for some time become magnetized. It is called induced magnetism or residual magnetism. So this should not be done in our nanoparticles. So this makes spions a unique tool in MR, magnetic resonance imaging, as a contrasting agent for type T2. What is the meaning of T1 and T2? I will show you in detail. So whatever I explain you, it is there in the slides. You can see a single particles. You can see a single particle having a net magnetic moment. You can see in figure B many particles having a net magnetic moment. If I have bulk particles in figure A, it will show a magnetic behavior. It is called ferromagnetic behavior shown in figure C. It is called hysteresis loop. But if I have a spions which show super paramagnetisms, this each particle show magnetism under a magnetic field only. And this, they will show a graph like sigmoidal graph. It can be seen in figure D. It can be seen in figure D. So it should show a super paramagnetisms. So sigmoidal graph, it is called VSM curve, is a signature of any magnetic material showing the super paramagnetic properties. Okay, now come to some principles of MR imaging or magnetic resonance. It is the extension of the concept of NMR. The students who are in chemistry, they very well know the concept of NMR. So it is the application of this NMR machine in a medical field. It is called MRI, magnetic resonance imaging. The concept is same. So what's happened here? You can see in the figure A, that the water proton is precise. Moving, precision means moving circularly in presence of magnetic field. You can see here in red font, net magnetic spin M of a water protons precise with a Larmor frequency W0. Okay, now what I am doing? I am applying the radio frequency pulse. It is called energy. I am applying the energy of this moving water proton. On application of this radio frequency pulse, the magnetic spins of this water is changed to the perpendicular directions. Okay, it will change in a perpendicular direction of the magnetic field. You can see in the figure B. Okay, now what I am doing? I am removing the magnetic field. Now slowly and slowly, this energized water molecule get relaxed. Agree? What I have done? I have keep the water proton in the magnetic field. They are precessing with the Larmor frequency. I am passing the energy in form of radio frequency to this water proton. Because of absorbing these radio frequencies, the precision of the water proton change to the perpendicular to the magnetic field. Now I am removing the magnetic this radio frequency pulse. As energy is removed, this excited water proton get relaxed. Now they can relax by two ways. First is called longitudinal relaxation T1 and second is the transverse relaxation T2. Students, you can see how this nanotechnology have a combination of all these three concepts. I am combining whole three: chemistry, physics and biology. So now see how they relax. They relax by two ways. If longitudinal, first graph. If transverse, second graphs. It is okay. This physics up to this, it is okay for you. You can see the graph slowly and slowly. These graphs, the graph shown in figure C, the bottom graph. It is a waveform. Slowly and slowly, this waveform becomes straight line. Now see in the next figure. It is a normal behavior of water proton in magnetic field and radio frequency pulse. Now what I am doing? I am inserting my nanoparticles, magnetic nanoparticles in the water system. Now water proton come in contact with the nanoparticle having magnetic properties. So water proton experience two types of magnetisms. First, applied magnetic field B0 and second, local magnetic field produced due to the nanoparticles. And because of these two types of magnetic field, now when I am applying a radio frequency pulse, see the relaxation procedures. It is very fast relaxation. In a first, see this. In a first, slow relaxation. In a without magnetic field, without local magnetic field, slow relaxations. With magnetic field, fast relaxations. So what is the importance of this? The relaxation determine the contrasting in the image. If fast relaxation is there, the signal get entrapped and I will have a very dark image. See here, dark image of water proton. Water, that is. If slow relaxations, normal relaxations, a weak signal. Fast relaxations, strong signal. Now one more technical term. T2. T2 is the time required for the transverse magnetizations to drop to 37% of the original value. I don't want to go to the technical term. Just I want to give you the message that shorter the T2, faster the relaxations, loss in signal intensity, and I have a darker signals. So nanoparticles, magnetic nanoparticles can produce a darker signals in MRI imaging. So in presence of magnetic nanoparticles, proton inside the local dipolar field perturbation area around the nanoparticles relax faster, produce stronger MRI signals. Again come. So why I require a contrasting in MRI? See, I am going to have a MRI of my body. Now doctor want to detect some disease area. So how can doctor detect some disease area? So the contrasting of a single disease area is required, which separate the disease area than the other organs. Now in case of development of cancerous nodes, the detection of spreading of this disease is very highly required. And for the purpose, contrasting is very required, very important. So I can compare this contrasting ability with the twinkling of the stars in the background of the night sky. There is a background of the night sky is there, and I can see the twinkling of the star very easily. Star can be my diseased lesion, my cancerous nodes, and remaining night sky is my body, like that. So my contrast agent should work on a particular area only. So what I am doing, I have a carrier with me. That nano carrier go to that particular cancerous cells, cancerous nodes. In that cancerous nodes, if it is collected, it is magnetic in nature. It will produce a contrasting effect in MRI, and also it will release the drug. So my carrier can do the dual job. It acts as a diagnostic agent. It is helpful to diagnosing the disease. It is helpful to map the cancerous nodes, and then it do the treatment of this cancer by releasing the drug from its cargo, from its carrier. From this carrier, drug is released for a target specific way, on a target specific way. So this type of combination of this technology is called theranostic agent or theranostic technology. Theranostic word coming from therapeutic as well as diagnosis. So diagnostic and therapeutic word combined from theranostic word. So this is called theranostic technology. It serves dual purpose. Now see the problem. Now problem start. I have just defined the requirement. What is the requirement for a magnetic material to act as a contrast agent? It should have a paramagnetic properties. Super paramagnetic properties. And super paramagnetic property is affected with the size. See this. When I reduce the size of my particles, you can see the figure B. I am reducing the particle size. There is a spin canting effect. What is the meaning of spin canting effect? The magnetic spin become delocalized, become disordered. And because of the spin canting effect, the magnetic property of a given material lost, which is not desired. You can see the property. Size, as size increases, canted surface spin decreases. See here. If I have more size, less canting, very strong imaging. But large size removed by the body. Large size is not desired. But if large size is there, spin canting effect is very low, very strong magnetic image. See here, with increase in the size, magnetic moment is high. If I am decreasing the size, magnetic moment become very less, not desired. MRI T2 contrasting agent effect. What is the meaning of T2 contrasting effect then? The MRI T2 contrast means it makes this contrast agent make the material, make the image dark than the background image. Get my point? It will make the image dark, make the cancerous nose as a darker image, and remaining material is a bright. So here MRI T2 contrast agent, this efficiency is very high with the larger particle size. If lower particle size, contrasting ability decreases. But see, this iron oxide is nothing but what? It is present in our body in form of hemoglobin. So this iron oxide is very, very non-toxic. See, I have asked the question to the first speaker of this conference. What is the fate of gold nanoparticles? Silica. They show some type of toxicity to the body. But what about this iron oxide? These iron oxides are recognized very well by our body. When their purpose is over, they are degraded by the enzymes, local enzymes, into the iron. And iron can be used to making the blood, red blood cells. It is called, this process is called erythropoiesis. So through the phenomenon of erythropoiesis, we can use this iron coming from the iron oxide nanoparticles. Okay. So these are the advantage, but particle size matter the most. Now, how can I control all this property? Now chemists come into picture. Now there is the role of chemist. See here. Okay. So this is about all about T2 contrasting agent. What is T2 contrasting agent? I have shown you this one. Stronger image, very, very dark object. Background is very white. Now let us have some look about the T1 contrasting agent in MRI. See, most of the radiologists and most of the contrasting agent available in the market is T1 contrasting agent. And this T1 contrasting agent make our lesion brighter. Lesion means target. Make our lesion brighter compared to the other objects. And these T1 contrasting agent, which are mainly sold in the market, are gadolinium based, GD metal based. And as you know, GD is a very, very highly toxic. But this GD metal is encapsulated, coordinated with the hexadentate ligands. With the hexadentate ligand, or even with a ligand with the 9 covalent sites. 9 means this GD metal. See here, you can see the figures. GD is all coordinated by oxygen and nitrogen atoms, and this ligand is called DOTA ligand or DOTAP ligands technically. And the 9th coordination valency is satisfied by the water molecules. This is the chemistry point of view. I am showing you some chemistry. So water is there. Water is coordinated as a 9th valency with these gadolinium metals. And this coordinated water has a major role, take into mind, to produce a brighter image in the normal MRI technique. So see here, you can see how this water molecule have a role. During the concept of MRI, I have shown you water proton persists freely in the magnetic field. And when I put some local magnetic field, its relaxation get differentiated. Here what's happened? Here in this case, water proton or even water molecule is bound with the gadolinium. And this bound water proton show a different magnetic behavior than the bulk water, non-bounded bulk outer sphere water proton. And in our body, we know that most of our body fluid made from water. So because of this type of two types of water, the contrasting effect is produced in the magnetic field. It is called brighter contrasting. It enhances the signals in MRI. There are a lot of physics involved. I don't want to go into that, but simply I can say that to produce a T1 contrasting ability, I require a bounded water proton. To produce a T2 contrasting ability, I require a water proton in contact with magnetic field, local magnetic field. Okay. Now come to the next point. See what's happened, how these two techniques can be combined. Such type of water molecules known as inner sphere water molecules are mainly responsible for contrasting effect due to the different longitudinal relaxation rate. T1 effect is produced by difference in longitudinal relaxation rate, and T2 is by transverse relaxation rate. So shorter T1, faster recovery, enhancement in the signal intensity. Shorter T2, faster loss of the signal, enhance, and there is decrement in the signal intensity. Produce a darker effects, enhancement in signal intensity, produce a brighter image. Okay. Now the idea was there. So see, in a traditional MR imaging using T1 or T2, both T1, T2 are available in the market. Most of the doctor using T1 contrasting agent based on GD metal. And we know that biologically GD metal is highly toxic. When these GD metal based complex are degraded enzymatically in the body, they release gadolinium. It directly affects the kidney and liver, and kidney failure is there because of the free GD metal in the body. It is called side effect of this contrasting agent. So it is happened there, patient go to the radiologist for diagnosing disease. Instead of diagnosing, he get the other disease like kidney failures because of the use of these contrasting agents. So leaching of the dopant metal ions from the host matrix was a major safety issue to be solved. So how can we solve this? Therefore, it is desirable to develop a contrasting agent free from GD ions, and any contrasting agent which also show the strong signal intensity in form of T1 or strong decrease in the signal intensity in the form of T2. See here. So if the idea was there, if I have inserting both the element, the T1 producing element which is GD or T2 producing element which is Fe3O4, combine it in a single material, then I will have a T1 T2 dual mode contrast agent. See here, you will have an idea. Considering the point, we can design the structure of the contrasting agent both that have both T1 and T2 modalities can be induced in a single materials. For example, R2 relaxation rate is called R2, can be achieved by controlling the particle size. We know that magnetic material affect the T2, GD based complex affect the T1. So T2 can be controlled by particle size. T1 can be controlled by inserting GD light complex, but not GD because GD is toxic. But this type of complex can be mimicked, can be copied. This working mechanism, this complex can be copied, and I can develop other systems which can have a mechanism same as GD. So see here, you can have an idea. This is a system. Here Fe3O4 nanoparticles act as a T2 contrast agent. I have a GD complex act as a T1 contrasting agent, and I can combine them with a spacer. So when they are present in the system, in the system, they can act both T1 and T2 based signals. See here, MRI machine, T1 mode. See here, brighter object. T2 mode, darker object. आपको MRI में क्या करना है? Switch on off ही करनी है. If you make the switch on, you will have a lighter object. You make a switch off, another mechanism, same system, darker object. So this will help the radiologist to map, to measure the predicting of the disease exactly. Okay, so how can this be done? See here, this gold is a T2 contrasting agent, gold color. Blue is a T1 contrasting agent. See the system, micelle. Core of the micelle is T2. Cell, periphery of the micelle become T1. Same system. Another porous material. The main framework of the material made from T1. And inside the pores, I can entrap the T2. I have a core cell type material. See the nanotechnology. T1 is a cell. T2 containing object is a core. Both T1, T2 are present. And where is the drug? In between of these, I can insert or enter my drug. So this system can act as a theranostic agent. I will put this in the body, in a system, and release it. When it go into the cellular, into cancerous cells, they release the drug, and they themselves act as a MRI contrast agent. You can see here the object. But the main drawback of this whole system is there. This T1 contrast agent is based on GD. So question is, how can we replace this GD? So I am in search of any contrasting agent which produce T1 effect without GD. Is it possible? Yes, it is possible. See here, I will show you how it is possible. But before that, let's have some introduction on the drug. In our study, we selected the leukemia drug. It is called imatinib mesylate. This imatinib mesylate was used for the chronic myeloid leukemia. It is a form of cancer of bone marrow. Cancer of bone marrow. It is called blood cancer. Due to the increased and unregulated growth of the myeloid cells and resulting accumulation of the blood. This occurs due to the translocation of the Philadelphia genes and something abnormal in the gene. Due to the genetic abnormality, this type of cancer is happen. The imatinib is a drug useful for this treatment of this. However, the treatment exerts many side effects like nausea, muscle cramp, rash. This is the side effect of imatinib. Abdominal pain, immune suppression. Worst is a congestive heart failure. So if imatinib is targeted directly to the disease site, maximum response with minimum side effect can be achieved. So what we have done? We have used Spion for this purpose. Spion can be simultaneously used as a vehicle for controlled drug delivery and as a MRI contrast agent like T2. So they can act as a theranostic agent in a true sense. So in that work, what we have developed, I have shown in that figure in the second slide graph. Now see, this is the method how we prepared it. It is very preparation of nanoparticle is very simple. In the lab, we can prepare it. I don't require any sophisticated lab. In a normal lab, I can do this FeCl, mix FeCl3, FeCl2 under acidic medium, then add EDTA, make a basic conditions. We will have FeOH twice or thrice, and then heat it at 110 degrees centigrade. Get hydroxide of Fe converted into oxides of Fe in form of Fe3O4. How can we say that the Fe3O4 is formed? We can simply apply a magnet. This magnet will show you the particle is just attracted towards the magnet. It will show you that the material is formed. And during this preparation, I can add the drug which is entrapped inside the micelles of the nanoparticles. Okay, now how can I say that it is Fe3O4, Fe2O3, it may be pure Fe. So X-ray powder represents the best one. Then how can I find the shape? By TEM. How can I find out the magnetic property? By VSM analysis. How can I find out the loading and unloading of the drug? By UV spectrophotometers. All these techniques are available in the normal laboratories. You can see this is the drug loading efficiency I have measured. When I loaded the drug during the synthesis, I found out with the help of UV how much drug was loaded inside it. And I confirmed that drug is present inside the synthesized system. Now see this XRD pattern. This XRD pattern is typically for Fe3O4. And these sharp peaks indicate that Fe3O4 nanoparticles are present in a pure form. There is no another phases of magnetic nanoparticles are there. And this spacing of the peak, the intensity of the peak and peak pattern prove that the inverted spinal structure is present. It is a chemistry point. This is a chemistry. Inverted spinal structure is there of Fe3O4. And this inverted spinal structure affect the property of the nanomaterial, magnetic property of the nanomaterial, which affect the MRI imaging. So I have to control the property of the material at the synthesis level only. It is called bottom-up technology of the synthesis of the material. Now see, this is the SEM image. This SEM image showing that there is no agglomeration. All small spheres can be seen. TM image even can be shown. These particles are monodispersed in nature. Monodispersed is a terminology useful for the particle size. If I have a much more uniform particle size, it is called monodispersed in nature. And all this lattice d-spacing can be seen. This indicate that my materials is highly in a nano domain and having a 50 nanometer size maximum. And they are highly crystalline in nature. This inset image is called the diffraction image, SAED pattern, selected area electron diffraction pattern. It shows that my material is crystalline in nature. This crystallinity of the material directly affect the contrasting ability. Now see, this graph. This graph indicate that the Fe3O4 nanoparticles are super paramagnetic in nature. On loading the drug, the super paramagnetic property remain intact, which is required for the contrasting agent. Now see this. Now how my EDTA, it is called ligand, interact with the Fe3O4 nanoparticle is very useful. So I can say that with the FTIR, I can say that how EDTA interact and adsorb covalently on the surface of Fe3O4. So this FTIR spectra prove this type of pattern. This type of pattern indicate that this red Fe3O4 nanoparticle remains suspended in aqueous medium, and EDTA make a cover on it, and two EDTA ligands are interconnected with each others, and this type of system is highly desired. Same as lipid nanoparticle system, here this Fe3O4 nanoparticle system remains stable in my medium. See, I am showing you how these techniques are useful to characterize my system. And now see the applications. I have used this material for the drug release purpose. In vivo study, in vitro study showing that the drug is slowly released in a buffer medium from this system. That means if it is not desired, if all the drug is burst at once, the slow release, control release property is also desirable. So this profile shows that the drug is released in a controlled way from my system. So this has an excellent sustained release properties. Okay. Now this is MTT assay. You know why this is required. This is so that this whole assembly is non-toxic. So treatment of K562 cell line. It is a bone marrow cell line with imatinib loaded nanoparticle cause an obvious decrease in the cell viability at 48 hours as compared with the pre-imatinib. Over 70% cell viability is maintained in the human this cell line. Okay, this IC50 value and PI staining shows that apoptosis death mode. This apoptosis death means controlling this all cancerous cells are dead. This cancerous cell become dead because of the effect of this drug in an apoptosis way. Necrosis was not favored. In vivo now, we inject our system to the mice, and we, after some time, after prescribed time, we have the analysis of the plasma, bone marrow, and liver. It can be seen here that this red indicate a sole nanoparticle, blank nanoparticles, and blue indicate a magnetic nanoparticles. So we can see that our system with drug, developed system with the drug, mostly collected at the bone marrow, while others with system or while in other organ, their concentration is very less. So from the uptake of cell uptake and cell viability study, it is observed that the drug-loaded nanoparticle was able to transpect the cell membrane and accumulate in the cell despite having a highly hydrodynamic radius. Compared to the normal tissues, tumors have, I explain you, leaky pores, and because of the large fenestration in the capillary walls that permit the passage of these larger nanoparticle systems. So these larger particle systems have their own advantage. In a normal cell line, they cannot enter because of the tight packing of the cells. So they will enter only and only cancerous cell line. So this is the advantage of this effect. EPR effect. The pore cutoff size of the porous blood vessel in the majority of the tumor is 380 to 780 nanometers. The maximum size of nanoparticle allowing penetration through the cell membrane is known to be 500 nanometer. So this is the demonstration, leaky pore EPR effect. In this, our nanoparticle can enter, system can enter easily. This shows that the cell line is not affected very much. It is safe. Now, I want to test it under MRI. So what we have done, we make a different vials having this system with a blank water, and we keep it under the MRI machine. Only our system in aqueous medium, and another system, another test tube, we have blank water only. We observe that our system work in a both the way. See, 0.001 milligram per ml is sufficient. 0.001 milligram per ml is sufficient to produce the T1 effect. See here. So we perfectly here use 0.1 milligram per liter is sufficient to produce T1. See here, T2 ke liye also it is 0.1 milligram per liter. You can see here. Water have no role, no disturbance, perturbation in between. Okay, so we can say that a contrast equipment can be achieved at a concentration nanoparticle as low as 0.01 milligram per ml. This is preferred for clinical use of the material as a contrasting agent to generate an appropriate signal with a minimum level of associated risk. Here, obviously, there is no risk. The developed system is very safe. Blank system is very safe because we have only Fe3O4. And you know this EDTA has some sequestering capacity. After some time, it starts sequestering Fe3O4 and converting this into Fe, which can be used in a erythropoiesis phenomenon. Okay, so this is a technical term. If I want to put the T2 weighted image, it is called retention time 3000 millisecond and echo time 99 millisecond. It is called T2 weighted image in the terms of radiology. T1 weighted image, I will make a repetition time 1000 millisecond and echo time TE 15 millisecond. So I can differentiate T1 and T2. See here, from this data, we have drawn a graph of retention rate versus concentration. So the synthesized system can act as a T1 and T2 both. From this graph, we can have R1 32.71. Just take it into mind. R2 295. Single system, Fe3O4 EDTA pluronic, R1 32, R2 295. Keep this value in mind. Now compare this with the commercially available. R1 0.22, R1 0.44 for this system. For other system, R1 1.2, R2 31.2. For lanthanide based system, R1 6.74, 778 R2. For other system, folate bovine serum albumin 196.1 R1 not measured, R2 179.5. See other system. Other people also develop spions. Generally, spions are useful for R2 relaxivity, T2 only. We and our group and very few people in the world were first to think about the T1 T2 dual mode. So in most of the time, this system was useful only for the T2 mode. So spion developed by other group 227, gold nanocells for T2 390, polystyrene based bead only 29.5. See our system 295.26 R2 and 32.71 R1. This is a very good results. If I have a commercially available system, so T1 contrasting agent, Rezovist is there, Peridex T2 contrasting agent is there. If we compare this, so our results are comparable. So we can say that our system work both T1 as well as T2 contrasting agents. Okay, so now the question is, now we bring this study for the imaging, actual imaging, in vivo imaging in a mice. We have just image of the mice bone marrow. There is a very blur image. Now we inject our system and keep it for 5 minutes, for 15 minutes, for 15 minutes, slowly and slowly, see, image contrasting producing. We take the image again after 24 hours, slowly and slowly only bone marrow can be seen. See, this study was carried out in a very, very old and primary MRI machine available in the General Hospital, SSC General Hospital at Vadodara. But we propose that if this system is useful for a sophisticated MRI, we can see or do the imaging with a very less concentration and very high efficiency. Now the question is, as you see, why this system act as both dual contrasting agent? Generally, the Fe3O based system act as a T2 contrast agent. They darken the image. But why our system work as a both? Why it's so T1 contrasting? If I have shown you in the image, for T1 contrasting effect, bound water molecule is required. If bound water molecule is present in the system, it is differentiated with the bulk water molecules under the magnetic field, and they produce the contrasting effect. So now we want to prove that whether the bound water is there in our system, which produce the T1 effect. We have done the thermal analysis of our system. In thermal analysis of our system, showing that, see here the red graph. Red graph show the two peaks. The peak before 100 and the peak after 100. The peak nearer to 100 degree is a normal water. And if the water is bound, obviously it requires some more energy to coming out. So second peak is observed. So we are successful to prove that both the type of water molecules are present in our system. You can see now here Fe3O4, EDTA, fluoronic. The outer is a bulk water molecules, outer molar molecules. Now you can see some entrapped water molecule present inside our system near the hydrophilic region of Fe3O4 core. Now when we burn it, first of all, the outer molecule, water molecule keep a peak below 100, and this water molecule requires some more energy to coming out, and because of that, second peak is generated. So we propose this figure, and our proposal is get accepted. Our paper was able to publish in a Royal Society of Chemistry journal. Our proposal was also accepted by the Department of Science and Technology, DST, SERB, and based on this work, our group was funded to do further research in this area. So here, we also use this system for a gene delivery purpose. We loaded gene delivery or gene, particular gene, separated from the E. coli bacteria. And we are able to successfully loaded this gene inside this system and carried out some study based on gene delivery. So on concluding, I can say that the method adopted for the immobilization of drug is low cost, practically feasible, sustained drug release could be achieved. If synthesis is carried out in an aqueous medium, then some water molecules can be entrapped with hydrogen bonding inside the system, which can produce a better T1 contrasting under MRI. Fe3O4 itself act as a T2 contrasting under MRI, and our system can be useful for dual contrasting agents. So we publish, based on this theme, we publish a paper in these journals, and then we are thankful to, I am thankful to DST, SERB India to funding this work. Even our university cell also funding this, UGC also funding our work. And this work, particularly what the work I have shown you, his work was carried out with one of my research students, now she is Dr. Ekta Shah. She is now a lecturer at our own institute, Polytechnic, MS University. These are the Faculty of Science. This is the Faculty of Science, Chemistry Department. You may visit this. This is the another image of Department of Chemistry. This is the image taken at 1 p.m. in the night. Our group used to work, particularly for a particular work, we have to stay in the night in the laboratory. So we captured this image, night image of our department. See, we are observing the protocols, COVID protocols. Offline exam is going on in our classroom. Okay, thank you. Any questions are welcome.
Thank you very much, sir, for sharing your knowledge and expertise with us. Session is open for discussion. Sir, we have collected some raised questions. May I, sir?
Okay. Yeah, thanks. Please go ahead.
Sir, what is the advantage of iron nanoparticles over gold nanoparticles?
See, gold nanoparticle, first of all, gold itself is very costly. And to synthesize, at the chemist point of view, if I want to synthesize gold nanoparticles, I have to take gold salt. It is AuCl3. It is very costly. Second thing, to convert this gold salt into gold metal, in AuCl3, Au plus 3 is there. Now I want to convert this into Au0 nanoparticles. So I require some reducing agents. And to have a particle size, monodisperse particle, I have to adopt some roots, it is called organometallic route for the synthesis. And organometallic route for the synthesis of gold nanoparticle is very tedious. It requires sophisticated laboratories. If I am adopting an aqueous route, for example, I can use ascorbic acid. I can use citric acid as a reducing agent to reduce AuCl3 into Au nanoparticle. But the particle size cannot be controlled. So it results into wasting of the salt, costly salt. But instead of this, if I want to make a Fe3O nanoparticle, I require a very cheap FeCl3 and FeCl2 mixing. FeCl is very cheap, FeCl2 is very cheap. Synthesis, just mix the two solutions, aqueous solutions, make the pH constant, make a pH constant, whatever basic pH required, first acidic and then basic pH. Then you will have this material. Very easy to prepare, very cheap raw materials. So these are the advantages. And the most advantage is magnetic in nature. If I want to see, before making any system, you should have an application of the system in your mind. So my application was to use this system as a MRI contrast agent. So gold cannot be used. Of course, nowadays reports are there. By modifying the gold in different way, I can use it. But here normally Fe3O4 are useful for the MRI contrasting effect. So if I want to use them as a CA contrast agent, so Fe3O4 is required. It is very cheap. Is it okay?
Yes, sir. Thanks, sir. Sir, one more question. As you said about iron nanoparticle loaded drug.
Yeah.
So is there any chemical bonding occur between iron and drug?
Yes. See, direct chemical bonding between the iron and drug is not preferable. See what's happened. When I am designing the carrier, the designing of the carrier is very important. During the designing of the carrier, we should have in our mind for what purpose we want to use them, which cancer we want to use them. Suppose I am directly load my drug on the iron without encapsulating it. It is possible that the drug depending upon the chemical nature of the drug molecules, it entrap it very tightly with the surface. If I am directly loaded the drug on the nanoparticles, it stick very tightly on the surface. So it is happened that when they go to the target, it may not be released. It may remain stuck with the material. What is our purpose? We don't want any direct contact between our nanoparticle surface and drug. So what I have done? We have done some layer on it, 2 layers on it, and between these 2 layers, the hydrophobic, the hydrophobic layer is generated. I can show you. This HLB balance is very important. Here you can see.
Yes, sir.
What is the use of this pluronic? One of the questions is why I require this pluronic polymer. It is a pluronic block copolymer. Can you see this slide, madam?
Yes, sir.
Number slide?
Yes, sir. Yes, sir.
It is a pluronic block copolymer. This pluronic block copolymers have 2 regions, red and blue. Red is a PPE, polypropylene oxide, and blue is a polyethylene oxide. This polypropylene oxide is hydrophilic in nature, so it remains towards the Fe3O4. And PEO is a hydrophobic in nature, it remains outward. Get my point?
Yes, sir.
Now this EDTA. This EDTA is make a first layer. See the red region around Fe3O4. This red region is hydrophilic. Red region is hydrophilic.
Yes.
And the blue region is hydrophobic. So we have a clear hydrophobic region between my polymeric micelles and EDTA. And this hydrophobic region is very important to attract the drug molecules. It is called hydrophobic effect. It is called hydrophobic effect because of the see most of the cancer drugs are hydrophobic. It is a major drawback. Even curcumin is the best one. Curcumin is haldi. Curcumin is the best one.
Yes,
But the
sir.
drawback of the curcumin is that when we take it, it is immediately released from the urine through the urine. It is not effective. Why? Because hydrophobicity is a major drawback. Our whole system, body is hydrophilic in nature. So this we have to control these properties. So what we have done? We entrap this drug in the hydrophobic area. See here, even in the second slide. See, in that second lecture, Madam has shown you. I have shown you just the concept because I am a teacher. See here, you can see these liposomes. This liposome, inside the liposome, see there are two types of regions: hydrophilic, hydrophobic. So this chain indicates the hydrophobic region. So if I am loading the drug, which is hydrophobic, it is entrapped in between. See this seat, bilayer seat. They are entrapped between the bilayer seats. And this hydrophilic area interact with our body parts. Get my point? So we are mimicking this natural system. See, madam told that these lipid particles, of course, they have their own advantages. But the disadvantage is there, these lipid nanoparticles are again very costly. They have their specific applications, as madam shown you. But in general applications, Fe3O4 is the best one because our system recognize it. Our system is knowing it. It is Fe3O4. And our mechanism in our body, erythropoiesis is there. Red blood cells are there. So they use this Fe. So this Fe on degradation go to the Fe pool in a process of erythropoiesis. Is it okay?
Yes, sir. Yes, sir. Thank you, sir. One more question, sir. Sir, should I continue?
Yeah, sure, madam.
Sir, what crosslinker you might have used for ZnFe3O4 particles coating with chitosan?
You see, this work was proposed by the other. This was the work of others. I have made our system. Chitosan and all these things are made from proposed by other. We have just the inspiration of this. All the explanation of chitosan is there, lipids are there. They have two types of surface. Inner surface are hydrophobic, outer surface are hydrophilic. And see here, the cross linkers are required to fuse the remaining two systems, like for example, my drug, and to make the drug and the system interact with each other, fuse with each other, we require some cross-linking agents. So depend upon the requirement of the systems, we have to select the cross-linking agents, the method of synthesis, and the raw material required for the synthesis. Is it okay?
Yes, sir. Mr. Raj, I think your doubt is solved. You can also unmute yourself and you can ask if any doubt is there. Mr. Raj Naik. Okay, fine. Sir, one last question. How drug metal compatibility is confirmed?
Drug and metal compatibility. Okay. See here. First of all, I want to show you. Just wait. I am not able to load again my. Just wait. Yes, this one. Yes. So if you want to create the drug metal compatibility, I have shown you in the very beginning. See here. Yes, this one. First of all, see. If drug disturbs, we have added the drug. We have added the drug during the synthesis of the material. During the synthesis of the material, nanoparticle system, in between we have added the drug. If drug acted some adversely with my materials, even some material act as a reducing agent. Instead of converting the Fe salt into Fe3O4, Fe2O3 may also form, Fe particle may also form, and they are all magnetic in nature. So how can I confirm that they are compatible? First of all, XRD. XRD, on making the XRD, if the pattern remains same with the blank, then I can say that there is no adverse effect. Again, another thing, magnetic property. This material is highly magnetically sensitive. If there is some adverse effect, the super paramagnetic nature will disturb. If super paramagnetic nature remains, it shows that, indicate that there is no adverse interactions between the drug and my material. And the third most important thing is that drug is not directly come in contact with my core. See here, the cores are all encapsulated first with the EDTA. You can see the slides, and these blue lines are there. These blue lines collectively form the hydrophobic environment in between, and this drug is entrapped inside this. Just take into consideration our figure. This figure is very important. See, our nanoparticles directly come in contact with the EDTA. EDTA is a very high affinity for Fe. Once it is covered with the EDTA, we make a second layer of micelle, polymeric micelles, and then we entrap our drug in between this hydrophobic area. So there is no question of interaction of my drug with the Fe3O4. And even if some area is left and drug interact with the Fe3O4, there is no adverse effect confirmed from VSM and XRD. Okay? Is it okay, madam?
Yes, sir. Yes, sir. Definitely. Fine. I think there is no more question. Thank you very much, sir, for sharing your knowledge on developing iron oxide. It was very informative session. We'll continue with the session. Now I invite Dr. Sandeep Chovatia to present vote of thanks.
Thank you, Nisha madam. Good afternoon, everyone. It's my privilege to propose a vote of thanks on this occasion. I, Dr. Sandeep Chovatia, on behalf of whole ARIBAS family and the entire fraternity of one-day national virtual seminar on nanotechnology, chemistry and biology at the interface of 21st century here together, and on my personal behalf extend a very heartily vote of thanks today. To begin with, I extend my deep gratitude to Engineer Sri Bhakti Patel Sahib, Chairman, Charutar Vidya Mandal and President of Charutar Vidya Mandal University for encouraging us to take up this event and provide all the required assistance. I am also thankful to Dr. P.M. Udani Sir, Director General, CVM University, for his continuous motivation. A big thank you to Dr. Bhakti Bajpai Madam, in charge at ARIBAS, for her wholehearted support in successful conduction of this event. I would also like to thank GUJCOST for financial support to conduct this one-day national virtual seminar. I am also thankful to our expert, Professor Pandian sir, Dr. Hemant Soni sir, and Professor Vanna madam for sparing their valuable time and sharing their knowledge to us. No event can be excluded, let alone be made on every success without gritty planning, imaging, and lot of determination which is required. I must mention my deep of appreciation for Dr. RItu Dixit, Madam, coordinator of this event, for her tireless effort in making this event a reality. I would also like to acknowledge my gratitude to Dr. Khushal Patel, sir, organizing secretary, for his tireless efforts in making this event successful. I would also like to thank our team members, Dr. Nisa Dakshini Madam and Mr. Sohil Patel sir, for their support and technical assistance. Last but not the least, I would like to thank you, all the participant students and teachers from different colleges, for making this national virtual seminar a successful event. Without them, it would not be possible. So once again, I thank them all for their participation. We are also grateful to our non-teaching staff of college for their cooperation. Once again, I thank to all the experts, participants, other faculty members. Thank you very much for your kind support. Thank you very much, Hemant sir.
Thank you, sir.
Thank you,
See you
sir. Thank you
again.
very much.
Thank you.
There are some instructions regarding certificate and declaration of results. The link for the feedback on the present national virtual seminar has been sent to chat box and WhatsApp group also. You are requested to submit the same to receive your certificate. Participants will receive certificate within couple of days as we are trying to provide hard copy of the certificate. For results of oral and poster presentation, that is already going on in the this session. Everyone is requested to visit ARIBAS website as we will display results on website within a day. So with this, the seminar has reached to its end. Hope the content covered during this session by the eminent speakers of their respective field certainly fulfilled your expectations to learn new things. Again, I would like to thank GUJCOST CBM University, expert speakers, faculty members, and all the participants for support and participation. On behalf of ARIBAS, once again, I thank you all for joining us today and have a great day ahead. Thank you very much, sir. And all.
Thank you. See you again.